our institution believes in imparting man-making, character building, and value-added education. The reaccreditation of the college as A grade by NAC and the status of college with potential for excellence conferred by UGC bears eloquent testimony of its unshifting commitment to quality. Dr. Ambedkar College is a truly college like none other, and it is not surprising that it has won a very special and unique place in the academic, extracurricular, and sports landscape of Nagpur city, Vidarbha region, as well as in the central India. The strength of college students is approximately 5,000. Dear students, we have assembled here for the one-day international webinar on a very important topic, that expert directives and experiences to develop global research career in biological sciences, which is organized by the Department of Biochemistry and Biotechnology of a college. I, Dr. Mrs. Pratibha Siriya, Principal of the college, welcome all the delegates, UG, PG students, participants, and research scholars for this international webinar. I would like to extend my warm welcome to all the eminent resource persons of the today's international webinar, Dr. Ansul Lokdarshi, Dr. Sunayana Mitra, Dr. Depanwita Pal, Mr. Sujeet Bhoite, and Ms. Snehal Raut. I appreciate the efforts taken by our faculty member, Dr. Mrs. B.M. Mehere, Head, Department of Biochemistry and Biotechnology, Dr. Devrat Begde, Dr. Utpal Dongre, Rohan Thavre, Pradeep Hirapure, Shweta Paranspe, and other faculty members for organizing today's wonderful international webinar. Once again, on behalf of Dr. Ambedkar College, Diksha Bhumi, Nagpur, I welcome you all. And I really appreciate all the eminent speakers who are interacting with all the delegates from USA. Once again, I welcome you all. Thank you, one and all.
Thank you, madam, for those inspiring words of yours and the encouragement that you always provide us with. Now, I will call upon the head of our department, Dr. Mrs. B.A. Mehere, to give her a welcome address.
Hello. Good evening and good morning and hearty welcome to all of you for today's international webinar on expert directives, experiences to develop global career research in biological sciences. I, Dr. B.A. Mehere, Head, Department of Biochemistry and Biotechnology, welcomes today's international guest speakers, Principal of our college, Dr. Pratibha Siriya Madam, Coordinator of the program, and all my dear participants across the globe. This international webinar is a panorama of scientific bonanza and will play a pivotal key to unravel and navigate the various scientific mysteries in global biological research. Today, we feel fortunate and privileged to have with us a versatile team of young budding scientists and researchers willing to share their expertise, experiences, and directives through this virtual scientific platform. The relevant research advancements and technologies will definitely satisfy the urge of scientific communities and help to face the challenges in life science research. I once again welcome you all and congratulate all the participants for enrolling in, and the entire organizing team for the success of this grand international event. Thank you all. Thank you once again.
Thank you, madam. With those inspiring and motivating words from our dynamic head of the department, madam, let us now navigate to the first talk of the webinar by Ms. Snehal Raut. This session will be moderated by Mrs. Shweta Paranspe, so I would request Shweta to take over.
Thank you, sir. Good evening to one and all. I, Shweta Paranspe, Assistant Professor, Department of Biochemistry and Biotechnology, take the privilege and I am honored to introduce today's first guest speaker, Ms. Snehal Raut. Ms. Snehal is now pursuing her PhD in Texas Tech University Health Sciences Center, USA. Her research mainly focuses on developing stem cell-based in vitro models of Human Cerebrovascular System to study Alzheimer's disease pathophysiology. Ma'am has 6 years of experience, many research awards, scholarships, and various international publications to her credit. She is expertise in stem cell biology, tissue engineering, in vivo techniques, data analysis, and various techniques in molecular biology. Today, Ms. Snehal will be highlighting on the topic, assessing the impact of Alzheimer's disease on the blood-brain barrier using induced pluripotent stem cells. With this brief introduction, I welcome you, ma'am, to take over the session.
Snehal, please unmute yourself.
Okay, am I audible now? Hello?
Yes, ma'am.
Can you hear me?
Yes, ma'am, you are audible.
Okay. Thank you for the kind introduction. I will, without a further delay, I will start with my presentation. And the title for my presentation is Assessing the Impact of Alzheimer's Disease on the Blood-Brain Barrier using Induced Pluripotent Stem Cells. Alzheimer's disease is the most common form of dementia characterized by memory loss and inability to perform everyday activities. Worldwide, around 50 million people suffer from dementia. It causes changes in the brain, such as accumulation of beta amyloid fragments outside neurons and twisted strands of tau proteins inside neurons. It is believed that the initial abnormalities in the folding of amyloid beta and tau proteins lead to cascade of events resulting in the neurodegeneration and cognitive decline. The image here shows the difference between healthy brain and Alzheimer patient's brain with significant brain shrinkage in Alzheimer patients. Alzheimer's disease is mainly categorized into two categories. First one is familial Alzheimer's disease and sporadic Alzheimer's disease. This classification is based on the age. So early onset of Alzheimer's disease is due to autosomal dominant inheritance of specific mutated genes. Late onset sporadic disease is more complex and more likely result from a combination of genetic and environmental influences. The majority of the Alzheimer's disease patients appear to be sporadic. Current research on Alzheimer's disease has primarily been focused on synaptic dysfunction and neurodegeneration. However, Alzheimer's disease is a slowly progressing brain disease that begins many years before symptoms emerge. Symptoms of Alzheimer's disease vary depending on the stages of the disease and worsen over the time as disease progresses. It is still not clear if amyloid beta accumulation or tau tangles are a cause or a consequences of neurodegeneration. Therefore, the research should be focused on the other aspects of the brain important for its function. And one such element is the blood-brain barrier. So what is blood-brain barrier? The brain is highly protected organ. It is protected by skull, cerebrospinal fluid, and other protective membranes. And one such protective element is the blood-brain barrier. The vessels that vascularize the central nervous system possess unique properties such as endothelial tight junctions uh, with the expression of specific efflux transport systems. These systems are important to maintain homeostasis in the brain. It provides defense against pathogens and toxins that might be present in the blood. Current models which are used for the Alzheimer's disease studies involve in vitro models, including cell cultures of primary immortalized cells, patient-derived stem cells, and some in vivo models, including mice and rats, with genetically designed to mimic Alzheimer's disease conditions. The limitations of these
Meher?
current, yes, sir? Can
Sorry
you
to
hear me, sir?
interrupt you. You are not sharing your screen.
Oh, I'm sorry. I thought I shared.
Not a problem. You can continue from where you have stopped, but start sharing the slides.
Yeah. I thought I'm sharing it. That's what I'm having some problems here. Can you see my slides now?
Yes, now it's visible. You can shift it into presentation mode.
Okay.
Yes. Thank you.
So, yes. Okay. Is it fine, sir?
Yes, Snehal, go ahead.
Okay. So the current limitations of the in vitro models or in vivo models available for the Alzheimer's disease studies are the blood-brain barrier display poor barrier properties, and that limits the study of pathophysiology of the disease. Animal model presently available for the study possess interspecies differences that act as a limitation in disease modeling. And whenever the drugs are being tested on animal models, it is very difficult to predict safety and efficacy of therapeutics in people. So our model is based on induced pluripotent stem cells. And to assess the role of selected mutations on the blood-brain barrier, we differentiated patient-derived induced pluripotent stem cells isolated from two patients diagnosed with the familial Alzheimer's disease with the mutation in preslin-1 and preslin-2 genes into brain microvascular endothelial cells. I'll tell you the details what preslin-1 and preslin-2 in the later slides. So as I said, my aim is to assess the role of preslin mutations associated with familial Alzheimer's disease on the blood-brain barrier function. So depending on the Alzheimer's disease pathophysiology, there are various hypotheses, and major hypothesis which leads to the clinical diagnosis of Alzheimer's disease is the accumulation of amyloid beta. We wanted to see if the preslin mutations generate amyloid beta peptides, whether it's a loss of function or gain of function. We first performed ELISA, which is a quantitative sandwich enzyme immunoassay, and quantified differential secretion of beta peptides. We observed significantly higher levels of amyloid peptides in both preslin-1 and preslin-2 mutated endothelial cells compared to the control. So from this result, it's confirmed that both the mutations are not leading to the loss of function. It is actually the gain of function. And these preslin genes are a part of gamma secretase enzyme complex, which is involved in the cleaving of another protein, that is amyloid precursor protein, which eventually leads to the amyloid beta accumulation. Next, we wanted to see whether this secreted amyloid beta from preslin mutated cells affects cell viability. And for this, we perform a live or dead cytotoxicity assay. It's a staining-based assay. Dead cells produce red fluorescence and living cells produce green fluorescence. And as you can see from our results, both the Priestlin-1 and Priestlin-2 mutated cell lines are showing few red cells along with green cells. So the amyloid beta secreted from these two cell lines has moderate or considerable impact on cell viability. Secondly, we performed another cytotoxicity assay to show the cell viability, and that is cell proliferation assay. In this assay, we observed a significant reduction in cell viability in the mutated cell lines compared to control. Next, we wanted to see how this amyloid beta secreted from the mutated cell lines impact oxidative stress, whether it leads to the generation of reactive oxygen species that is leading to the cellular death, or it's maintaining the reactive oxygen species levels in the cells. From our results, we observe that there is an increase in the reactive oxygen species from Priestlin-1 mutated cell line, but there is non-significant difference in the Priestlin-2 cell lines compared to control. We confirm these results by performing another assay in which we used a immunofluorescence analysis using cell rocks. So these cell rocks shows increased ROS levels in the endothelial cells carrying Priestlin-1 mutation, and these released reactive oxygen species in mutant cell life leads to progressive oxidative damage in the blood-brain barrier integrity. So how these oxidative stress leads to the affects the proteins available on the blood-brain barrier, or how it affects the integrity of the barrier. For that, we perform immunofluorescence analysis, and we looked at the expression of tight junction markers present on the blood-brain barrier. So first marker is the Jc1. As you can see, the control cell line shows very continuous tight junctions, but there are no tight junctions present in the Priestlin-1 mutated cell lines. And this is also confirmed with the quantitative analysis. Another marker is a Claudin 5, which is also compromised in the Priestlin-1 mutated cell lines and also lesser immunoreactivity in Priestlin-2 compared to control. Next, changes in the barrier function was assessed by measuring changes in transendothelial electrical resistance and fluorescent permeability. One of the important characteristics of the blood-brain barrier is a tight junction and selective permeability to the molecules. And as expected from our earlier results, Priestlin-1 endothelial cells showed impaired barrier function compared to Priestlin-2 and control. As we can see, there is a significantly lower tier and higher fluorescent permeability in these cell lines. Next, changes in the glucose uptake and glucose metabolism in the Ipsc lines were assessed. Blood-brain barrier is highly expresses GLUT1 transporters, and from our immunofluorescence analysis, we saw reduction in the GLUT1 expression on Priestlin-1 mutated cell lines. How this downregulated expression of GLUT1 leads to the changes in the glycolysis or changes in the cell metabolism that we have carried out in the further experiments. Another major characteristics of the blood-brain barrier is the expression of efflux transporters. And as you can see here, there is the efflux transporter P-glycoprotein is severely impacted in Priestlin-1 mutated cell line. Also, there is less immunoreactivity in the Priestlin-2 cell line compared to control. Further, we wanted to see how these mutation or how the amyloid beta secreted from the mutated cell lines affect expression of the genes involved in the blood-brain barrier function and integrity. And from mRNA expression corresponding to the tight junction proteins like Jc1, flaudine, occludine, and efflux transporters such as GLUT1, BCRP, MRP, and some of the glucose transporters, we can see that there is upregulation of majority of the genes as observed by real-time PCR. In correlation with the protein expression, there was decrease in the GLUT1 mRNA levels that was seen in both the cell lines. These upregulation of mRNA expression indicates post-transcription modification and genetic variability in the cell lines. Next, we wanted to see how the low glucose or down regulation of GLUT1 or differential expression of gene mRNA and genes affects mitochondrial function. And to see that, we perform Jc1 staining, and this Jc1 staining gets accumulated and form J aggregates in healthy cells characterized by red fluorescence in the mitochondria. And we can see that control is showing J aggregates with the red fluorescence. During apoptosis, these J aggregates transform into monomers, leading to the loss of red fluorescence. And from our results, we can see that the preslin1 mutated cell line is showing more of green fluorescence, which is showing the dysfunction of mitochondria. And to further investigate the impact of such impaired mitochondria, we observed changes in mitochondrial function using cell flux analyzer. So this cell flux analyzer gives you the better idea about the cell metabolics, including glycolysis and mitochondrial respiration. So from our detailed analysis, we saw decrease in the ATP production. Yes. Yes, sir.
Sorry to interrupt you. Conclude within 2 minutes if you can.
Okay. Yeah. So from these data, we are seeing that there is decrease in the mitochondrial respiration that was also confirmed with the Jc1 fluorescence ratio. Another thing we wanted to see whether it affects glycolysis, and from our results, we saw there is decrease in the glycolysis because of the mutation. So to summarize, our results suggest that the mutation, preslin mutation, leads to the adversely affects the blood-brain barrier integrity and functions. And this disease model from patient-derived cells can enable the study of genetic variability with different patients. So with this, I would like to thank my mentor, Dr. Alamat, and the Texas State University Health Sciences Center. I would also like to thank Dr. Ambedkar College, Nagpur, Principal Ma'am, and respected other professors. And I would like to thank Dr. Devras sir for giving me this opportunity. I'm grateful, and thank you so much. And I can answer the questions if you want. Thank you. I'm sorry, I cannot
Shweta, you
hear
need
you.
to unmute yourself.
Okay. Yes, sir. Thank you, ma'am. That was an explicit presentation. I'm sure that all the participants are definitely benefited with this talk. As there are, I didn't see any questions in the chat box. So, ma'am, can you
Maybe
please
I can ask one, Shweta, if you can allow.
Yes. Yes, sir.
Okay, Snehal. It was quite a pretty interesting study, but let me tell you, I'm also interested to certain extent in Alzheimer's model. I'm working with Drosophila Alzheimer's model, as you might be knowing, or you might gather what I did at CCMB. So maybe related to that, is there any correlation between hypoxia and the stem cell architecture that affects during Alzheimer's? Can you correlate?
Yes, so actually, there is a correlation between hypoxia and Alzheimer's disease. I haven't seen the relation between that, but we have seen the relation between hypoxia and ischemic stroke. So we do have a plan of seeing, including hypoxia or oxygen glucose deprivation, and like as oxygen and glucose, these are mainly like these are very important for the development of brain. I think this both these factors will impact the development of the blood-brain barrier and its function. So yeah, I think you can include.
And one more I have for you. Uh, Blood-brain barrier has been shown to get affected by certain peptides, especially those which are secreted by the gut microbiome. Anything regarding that? Any comments on this?
I do not have an idea about gut microbiome, but I feel if these peptides are leading to some sort of cytotoxicity, whether it's a generation of reactive oxygen species or whether it's affecting the integrity of the blood-brain barrier, or if or any other mechanism, because Alzheimer's disease is like very complex and involves like a lot of factors. Yeah, so maybe even the gut microbiome or the peptides which are able to cross the BBB are getting accumulated in the inside of the brain and not getting cleared out because of the downregulation of efflux transporters. So that might lead to the impairment of the blood-brain barrier.
Snehal, I have a question. Actually, Dr. David.
Perhaps Sujeet has to say something about it.
Yeah, Dr. Devraj stole my question. I was actually going to ask you about the gut microbiome and its impact on the blood-brain barrier, because that's something I'm really interested in and study. But anyway, the second question is, we are talking about familial mutations in Alzheimer's, right? What percentage of people who actually suffer from Alzheimer's are actually suffer Alzheimer's because of familial mutation? Probably there are more sporadic Alzheimer cases rather than genetic mutations, right? Am I right in thinking that?
Absolutely. You are absolutely right. There is like 1 to 5% of patients in the familial Alzheimer's disease category, and with 90% of the patients are in sporadic form of Alzheimer's disease. And I'm actually studying both. I have prepared a model for both the studies, and I'm seeing similar effects, like similar modulation of genes, similar disruption of the tight junction markers present on the blood-brain barrier. And I'm comparing both, and I'm coming to the conclusion that maybe the GLUT1 or the glucose availability that you have to focus on for the therapeutic purpose, because the GLUT1 gets downregulated in both familial as well as the sporadic form. That is the major finding of my work, I can say. So my further or future studies will be focusing on the GLUT1 transporters and glucose uptake.
Thank you. Wonderful. Thanks.
Thank you.
Yeah, over to Shweta. Take over, Shweta.
Yes, sir. We have one question from the chat box by Somesh Divte. He's saying, is there any correlation between the biological clock or Alzheimer's disease?
Actually, the biological clock sleep cycle, it, as I said, it's a multifactorial disease. So many factors involving, including the environmental factors, genetic mutations, your diet, your biological clock, your circadian rhythm, everything affects at certain extent on the blood-brain barrier, on the neuron communication, because if you are deprived of the sleep, I don't think there will be proper communication between the neurons that will not be delivered to the other organs of the body. So yeah, obviously, I think there will be effect of your biological cycle on the disease.
Okay. Thank you, ma'am. Can I request for your email ID?
Shweta, just a moment.
Yes, sir.
Snehal, can you, going with the theme of the webinar, can you within 1 or 2 minutes tell all our participants how was your journey from, say, Nagpur to US?
Yes, I'm sorry for not including slides for that, but I'm happy to share my journey. It started from, I'm not from Nagpur. I came to Nagpur for my, after 10th standard, and I studied in Shivaji Science College. And as like every Indian family, all the parents, Indian parents, they want their kids to be a doctor or an engineer. So I also had that same aspiration, but unfortunately I could not become a doctor, but I wanted to have that doctor in front of my name. So what is another way to get that doctor is doing a PhD. So that's how I started my journey. I got admission in the BSc program in Islab College Biotechnology, and there I got first experience of working on any research project. And thanks to David sir, thanks to other respected professors in his lab school, they gave me opportunity. They provided me with that opportunity. And during summer vacations, I try to work or I try to have multiple internships in different research institutes. So I worked in Mehere. After my master's, I did my master's project in NCL. So from there, my journey started. I tried for another national entrance test that I could not clear, but I could clear the CCMB Research Institutes exam, interview, and other research institutes exam. And from there, I was looking at other students who were preparing for the GRE, TOEFL, and I thought, okay, I can also give it a try. And from there, I started preparing for these exams. I gave interviews. I looked at different universities from different countries. I applied to the professors, and yes, I was lucky enough to get selected for the PhD program in Texas Tech, and that's how I started my journey.
Shweta, over to you.
Yes, sir. Thank you, sir. Nice to hear it, these things from you. I'm also from Hislop College and a student of Devrat sir. Ma'am, can I ask you your email ID, because some of the participants are asking for it. Maybe they have to ask some questions or queries later on. Hello? Am I audible?
Yeah, yeah. Can you hear me, ma'am?
Yes, yes.
Okay, my
Yes,
email,
yes.
I will write it in the chat, so you can just copy it.
Okay, fine. I can share that email ID with the participants. No problem.
Okay.
I just wanted a nod from Snehal for going ahead with giving that to the participants.
Yeah, I'll be happy to answer their questions or help them in any way possible.
Once again, I thank you, ma'am.
Thank you.
Now I request Mr. Rohan Thavre, sir, to please proceed for the next session.
Good morning, speakers. Good evening, all the participants. Am I audible?
Yes, sir.
Yes, Rohan, you are. Go ahead.
Oh, thank you, Shweta ma'am. It's my pleasure to introduce to you our next speaker, Mr. Sujeet Bhoite. Mr. Sujeet Bhoite completed his undergraduate studies in zoology from Pune University in 2008 and postgraduate studies in biotechnology from Nagpur University in 2011. He got years of experience in biotechnology industry like Swasti Agro and Bioproducts Private Limited and Tata Chemicals Limited Innovation Center. In 2011, he co-founded a web-based venture, powerof9.com, which helps connect nanotechnology industry stakeholders to promote knowledge sharing. Currently, Mr. Bhoite is working as project manager and consultant at MI Leeds Consulting University of Michigan, and also pursuing PhD in molecular, cellular, and developmental biology, University of Michigan, where he is investigating the role of human gut bacteria in Parkinson disease causation. With this brief introduction, I request Mr. Sujeet Bhoite to address the gathering. Sir, over to you.
Hey, Rohan, thanks a lot. Good morning and good evening, everyone. I'm just gonna give me a second. I'm gonna share my screen with you so that yeah, this is it. Just give me a thumbs up if you can see my screen. Is my screen visible to all?
Yeah.
Cool. Thank you.
Yeah,
thanks,
it's
Rohan.
visible, Sujeet. Go ahead.
And thanks, Dr. David, for organizing this and giving me a chance to speak to so many great people and listening and learning from them. And it's a wonderful opportunity. And I've titled this talk like Following the Gut Instinct from PhD to Parkinson's disease, because I study the impact of gut microbiome on Parkinson's disease. But at the same time, the gut instinct, as we call it, also is something which propelled me to leave whatever a very stable job in Tata Chemicals and start pursuing PhD. So taking a cue from what Snehal said, I'm just going to give you a brief introduction about myself and what I do and how I got here in the first place and how you can get here at University of Michigan, Ann Arbor. And I'll help you and guide you through that process in just briefly. So about me, as I mentioned, I was born and brought up in Pune. I'm the first generation graduate student. That is to say, neither my mom and dad went to graduate school. So that was a big leap for me. And they supported me in all these ventures and helped me get here. So big shout out to them. With that, I did BSc in zoology from Pune University. And then I took a year of gap after my bachelor's in zoology. I caught snakes, worked with the snake park in Pune, did a lot of outdoor adventures things. And then I said, okay, probably now I need to get back to education studies. So I fortunately, I got into the biotechnology master's program at Hislop College, Nagpur. That's where I met Dr. Devrath. And Dr. Devrath is a person who actually introduced me to bacterial biofilms and the protein which make the biofilms in bacteria, especially in E. coli. And I was so fascinated by it that even now I study that in my PhD, but I'll come to that later. After my biotechnology, as mentioned, I started this website, a nanotechnology networking website called Power of -9 to get together the different stakeholders in nanotechnology, enhance the knowledge sharing between the industries and the scientists and the students working in nanotechnology, and help the commercialization of this technology, because I've always been a proponent of taking the science from the labs to the masses, to the people, and commercializing it. Unfortunately, that venture didn't last long, but that gave me a breadth of experience, which definitely helped me in my PhD and my current position here. Yeah, I did work with a small company as a startup company. It's called Swasti Agri and Bioproducts as a production manager. After a short stint over there, my major research happened, or a major career break was in Tata Chemicals, where I spent like 3 years of my life. I got 3 patents over there. I commercialized the product, and it was a well-paying job, but something told me I'm getting into a comfort zone, just doing the same thing every day and good paying job. And so I followed my gut instinct and said, I resigned one day and I said, I'm leaving because I want to pursue PhD in US. And that's what got me here. So I applied to a lot of universities. I got into a couple of them, but I decided to pursue PhD in University of Michigan. And at the same time, I'm also working as a project manager with my lead consulting group. And I have an asterisk over there because if we have time, I can definitely talk about it in future or in the later slides if we still have time. But currently, I'm pursuing PhD at the University of Michigan. The University of Michigan in Ann Arbor is you should probably consider applying to this university, or you should at least consider whatever program you want to apply, because this is #1 public university. We have 19 different colleges and schools, 250 plus degree programs, and 1,600 plus student organizations. So there's a lot of things to do extracurricular as well as, with respect to degrees. And the alumni network is really good because after you graduate, probably after you get your degree, you need to find job, and the alumni network definitely helps in that. And there are some really well-esteemed alumni at University of Michigan, which will be more than happy to help you out with your job search or networking, or getting a postdoc if you want to go for a postdoc. So that alumni network is very strong. So definitely do consider looking into University of Michigan and all these organizations and schools and colleges and schools we have at the University of Michigan. One of the department which I'm on is called the Molecular, Cellular, and Developmental Biology Department. So on the left, I've just shown you the total enrollment. This is from 2015 to 2019. As you can see, 60% or more of the students admitted to my program, this is a PhD program I'm talking about, are international students. So this is definitely more international students focused program. And more female students are. So it's also not only it's friendly for international students, also we have more female students than male students in our department, as you can see by this graph. So I'll definitely would encourage students who are aspiring to get a PhD or some master's program in US to definitely enroll or apply rather to my department, because I talk to many people in my department, mostly admissions committee related people, and one of the things they have mentioned is like they do not see many applications coming from India to our department, which is a big bummer because they really want more people from India to apply. Yeah, there's an application fee definitely, but I think it's still worth to try it because they want more applications. Mostly we see applications from Southeast Asia, China, Korea, those kind of applications, but they would definitely like to see more Indian people applying for that department. And I'll share my email and everything so that, if you have any questions related to applying to my department, you can definitely, send me and shoot me an email and I'll help you out. In my department, we have 38 different labs to choose from. In the first year, you will do a couple of rotations. You will just test out different labs, what they are doing. You'll do small a month or a couple of months of work in every lab or whatever labs you choose. And then you get to decide which lab you want to work in. And the model organisms is like you can work in bacteria, plants, xenopus, you can have C. elegans. We have diverse variety of model organisms and projects you could be a part of. And again, student organization is something we like to be very proud about because our students are not only doing biology outreach or on diversity committees, which our student organization has. Our students are also part of admission and recruitment committee. So our students actually look at your applications, and we are also very integral part of accepting students to our PhD program. So definitely consider applying to our department or at the University of Michigan in any department you want, and shoot me an email, which I'll share my email address. If you have any questions or you want to chat or have a Zoom call or anything, I'll share my details with you guys. With that, I will switch gears and talk about my research. I've made a very basic presentation, taking into account the limited amount of time we have, and talk about the gut instinct, or essentially how the bacteria in the gut could affect Parkinson's disease, or how bacteria in the gut and what is in the gut which could lead to Parkinson's disease. So we are calling this polymerizing the fiber between bacteria and Parkinson's disease. Why I call it, I'll tell you shortly. But just to give a brief introduction of what Parkinson's disease is. Parkinson's disease is a amyloidogenic aggregation of a protein called alpha-synuclein in the neurons. The major pathophysiology with Parkinson's disease is severe motor dysfunction. What happens is what happens is there's a protein called alpha-synuclein, which is random coiled in the body. It misfolds for some reason, and that starts aggregating and forming these amyloid fibrils. And these amyloid fibrils deposit in the neurons in the form of Lewy bodies. And these Lewy bodies are toxic to the neurons, which leads to the death of neurons. And mostly the neurons which are affected are dopamine producing neurons, which control the motor functions in the human body. So due to this motor dysfunction, there are different symptoms associated with Parkinson, which involves the shaking or postural instability and those kind of memory loss also. But there's an interesting thing that there are, with respect to the non-motor symptoms, there are also some with respect to the motor symptoms, there are also some non-motor symptoms which precede the motor symptoms by years. What I mean by non-motor symptoms is symptoms which are not associated with the body's posture or the movement of the arms or the limbs. These non-motor symptoms are basically mostly constipation, which is gastrointestinal dysfunction and constipation. So nearly 80% of the patients who suffer from Parkinson's disease complain about constipation, and that constipation actually precedes, or this gastrointestinal dysfunction occurs like 10 to 20 years before actual Parkinson's disease is detected in those patients, which leads us to question the early onset of these gastrointestinal dysfunction in Parkinson's disease probably suggests a potential gut-brain link here, because we all know the gastrointestinal functions of a human body are very also very affected by the gut microbiota. And if those are going haywire, or those gastrointestinal dysfunction are happening in a Parkinson's disease patient, probably there's a link between the gut and the brain in this case. So, and we are not the only people who have thought about it. Many people have actually looked into the gut-brain axis in the Parkinson's disease. What happens is alpha-synuclein fibrils, that is the protein which aggregates in Parkinson's disease. So if you inject the alpha-synuclein fibril into the intestinal cavity here in a mouse model, let's say, these alpha-synuclein fibrils are actually transported from the intestinal, where you inject them in the mouse, to the brain via a nerve called the vagus nerve. So vagus nerve is the nerve which connects the central nervous system of humans to the enteric nervous system, or the nervous system which lines the gastrointestinal cavity. So it was interesting that something you inject over here in the intestinal cavity is getting transported to the brain via this vagus nerve, which again suggests the gut-brain axis here. Secondly, there was this landmark study by a scientist called Brock, and Brock's hypothesis is like probably some unknown pathogen in the gut could be responsible for initiation of sporadic Parkinson's disease. As Snehal mentioned, since 1 to 5% of people have familial mutations, which leads to Alzheimer's. Similarly, in case of Parkinson's also, sporadic Parkinson's disease account for approximately 90% of the patients. Only 10% of them have some genetic mutations, familial mutation which leads to Parkinson's disease. So that's where the Brock hypothesis comes in. Probably there's some pathogen in the gut which could create some toxins which then affect something in the brain, which could lead to initiation of Parkinson's disease. And secondly, we in collaboration with the Caltech University have actually showed that human gut microbiome, if you take the gut microbiota from a Parkinson patient, human Parkinson's patient, can enhance motor and transplant that human gut microbiome to a mouse model. In that mouse, you can see enhanced Parkinson's disease, or the mouse model shows motor dysfunctions, which kind of tell you that there's something in the human microbiome or the gut microbiota which is causing these mouse to develop Parkinson's. So we always knew that we were working on this, and my lab is a biochemistry focused or bacterial focused lab. So in our lab, we study alpha-synuclein because it's a protein which aggregates and forms amyloid fibers. But at the same time, we also study a functional amyloid called Curli. So this is the amyloid I was talking about. This is the protein I was talking about, which Dr. Devrat introduced to me when I was in my doing my master's in biotechnology in Nagpur at Hislop College, and I was fascinated by this protein. What is this protein? This protein is called Curli or Curly, and here I've shown an E. coli cell, and these fibers are proteins. This is a Curli protein. So what happens is the E. coli cells or many enterobacteria cells make this protein called CSGA. And the CSGA is the major component of this. These fibers are nothing but monomers of CSGA stacking upon each other, forming these fibers. So the protein CSGA is made in the cytoplasm and secreted to the periplasm in the E. coli. And there's this protein pore called CSG, which is a 9-mer protein, and it is secreted outside into extracellular space. Once CSGA, as shown over here, is unfolded, random protein is secreted outside, it then starts polymerizing on the surface of the E. coli cells, helped by other proteins like CSGF and CSGB. But I won't go into it because we are more concerned about CSGA. And what happens once this protein is secreted outside, it starts polymerizing on the cell surface like this. It starts from a random coil, it assumes this beta sheet structure, which starts aggregating and forming these beautiful fibers. Why are these fibers important for the bacteria? These fibers are important for the bacteria to form biofilms, protect itself from moisture lost or protect itself from, if it is forming a biofilm inside the intestine, it helps itself from, helps the bacteria from human human immune cells and whatnot. So this protective layer is important. This protective protein called curla is really important for E. coli to form biofilms. In our lab, we purify CSGA in a test tube. We can purify it outside a bacteria and study its aggregation, study this protein thoroughly to understand what exactly this protein is. So if you can purify E. coli CSGA, and put it in a test tube, you can study how from a random protein, from a monomer, it starts forming aggregates to form these beautiful fibers, which we see under a transmission electron microscope. And how we study this is by an assay called thioflavin T assay. So this is a thioflavin T assay. What is thioflavin T, as THT as mentioned over here? It is a dye. This dye binds to these fibers. This dye has very low fluorescence intensity by itself, but when it binds to these fibers, its fluorescence intensity redshifts. So its fluorescence intensity, there is change in its fluorescence emission. And we can actually monitor that fluorescence emission and quantify the degree of fiber formation. If there's more fluorescence redshift, more fluorescence emission, then we can say there's more fiber formation. So as you can see over here, on the x-axis we have time, and on the y we have thioflavin T, THT fluorescence. As expected at time point 0, there's not much increase in the THT fluorescence because there's nothing much happening. But approximately around 2.5 hours, what happens is the E. coli, CSGA, the protein, starts forming these aggregates. The monomers start coming together and forming and sitting upon each other, stacking upon each other, forming these fibers. And now you can see an increase in THT fluorescence gradually. So this is the lag phase. If you are familiar with bacterial growth curves, you'll understand this is a lag phase when nothing much is happening. It's just building up, building up, building up. And then there's a growth phase wherein you start seeing a rapid increase in the THT fluorescence because the fibers are growing bigger and bigger and bigger. And once all the proteins in that solution, in the test tube, have all formed this aggregate, now you see a stationary phase because there's no more protein to add on to the fiber. So they have reached the stationary phase. So this is a typical THT fluorescence curve. And this is a curve I'm going to talk a lot about. So in my following slides. So this is very important. This is my gold standard assay. This is how we study the aggregation of this protein. What is happening in the lag phase is really important. What happens in the lag phase is that it's not that lag phase is a stage, nothing is happening. In fact, there's a lot of things happening in the lag phase. E. coli monomer, CSGA monomers are coming together and forming smaller aggregates, which gradually grow, and you can start seeing them over here. But there's an important property of most amyloid fibers is that if you take this time, if you take fibers, if you take the protein from this, at 24 hours, you take a protein and you add a small amount of that protein back here and you start that assay again, what happens is something called seeding. Seeding is something which is a unique property of amyloid fibers. So you take preformed fibers and you chop them into smaller parts. You can sonicate them and do different things. You can chop them into smaller parts and add only 10% of that back in this assay. And what you see now is that there is no lag phase and that protein immediately starts aggregating. What is happening is that instead of in the lag phase, when I told you that E. coli is CSG is forming those oligomers or those smaller aggregates, which then build into bigger aggregates. By adding a small amount of preformed fibers as seeds over here, you have gotten rid of this phase because you've already provided them with small aggregates. And now the protein in this test tube can immediately start aggregating. That's why you don't see any lag phase. You start seeing immediately a growth phase because you've already added you already added the seeds, or you already added the smaller aggregates which were needed to start the growth phase. In the normal E. coli, you have already added them by adding a preformed fiber. So this aggregation of CSGA in vitro in a test tube can be accelerated by adding a preformed CSGA seeds. We call them seeds because they're acting as seeds upon which the fiber starts building itself. This process is called seeding, and this becomes very important because the same seeding can be seen with respect to Parkinson's disease in a protein called alpha-synuclein. I've shown on the thioflavin THT curve over here. Again, this is alpha-synuclein by itself in blue. So you can see alpha-synuclein is a slow aggregating protein compared to E. coli CSGA, because E. coli CSGA starts aggregating in 2.5 hours. Alpha-synuclein takes like 15 to 20 hours to start aggregating. And it also displays the lag phase, the growth phase, and the stationary phase. If you run it longer, you can see the stationary phase. And just like E. coli CSGA, if you add preformed seeds of alpha-synuclein, small amount of preformed seeds to a monomer solution of alpha-synuclein, you get rid of the lag phase and you immediately start going into the growth and the stationary phase because you've already added the seeds needed for it. If you look at the TM, like shown over here at the right, there's not much difference, morphologically, there's not much difference between the alpha-synuclein fibers or the E. coli CSGA fiber. So they look the same. Which led us to a question, which we asked a question since we're studying these two proteins in the lab. Could there be some interaction between bacterial protein like CSGA and alpha-synuclein? Because both of them have a amyloid-like characteristic, both of them start aggregating, and both of them have display the same characteristic. So we thought, why not we add them together and see what happens? So that's exactly what we did. We took alpha-synuclein by itself, as shown earlier, and then it has a lag phase. Lag phase is the time it takes to start aggregating. We quantified it here. It has a lag phase of approximately 19.5 hours, as shown over here. And then what we did was we added a small amount of E. coli CSGA to it, a very small amount. So this is 50 micromolar of alpha-synuclein. This is only 2.5 micromolar of CSGA, which is just 5%. And what we see was an accelerated aggregation of alpha-synuclein. Remember, alpha-synuclein aggregation is one of the reasons Parkinson's happens. At least alpha-synuclein aggregation is seen in Parkinson patients. So here what we saw was there's a bacterial protein which is accelerating the aggregation of alpha-synuclein, which causes Parkinson's. Aggregated alpha-synuclein causes Parkinson's, so which was very interesting because this is the first time we have shown that a bacterial protein could affect the aggregation of a human protein and which causes disease. But this was E. coli CSGA. So the question we asked ourselves was, are there other CSGA CSGA proteins in the microbiome of human microbiome? Are there organisms which are aggregating CSGA like proteins in the microbiome? So what we did was we looked at the sequence of the human microbiome. And there's a database wherein you can look at the sequences of different gut bacteria. And from that gut bacteria, we isolated a couple of hundreds of different bacteria which make CSGA like proteins. In the interest of time, I'm going to talk to you about only two of these. The first organism which we isolated from the gut and which makes a CSG-like protein is called Hafnia. Hafnia is the name of the organism, and it makes a CSG-like protein. And we cloned it. We took out the gene. We cloned it. We purified the protein, which Hafnia. We call it Hafnia CSGA, and it also displays the same aggregation like kinetics, wherein it has a lag phase, a growth phase, and then eventually it plateaus off. And the second one we isolated was Citrobacter. We all might have heard about Citrobacter. It's also found in the human gut. And it also makes a CSG-like protein. So we purified Citrobacter CSGA too. And it also has a different aggregation kinetics. You can see it is fast aggregating, this is slow aggregating. Morphologically, they do not look very different. You can look the fibers on their TM and they look the same. So we did the same thing. This is the lag phase calculation. I just wanted to tell you that E. coli CSG has a lag phase of 2.5 hours. It's a relatively fast aggregating protein. Hafnia is a very slow aggregating protein, and Citrobacter is very fast. So remember, Citrobacter is a fast aggregating protein. Hafnia is a slow aggregating, and E. coli is an intermediate. So then we looked at the crosstalk. Just like E. coli CSGA, we asked whether these Hafnia CSG and Citrobacter CSG could it accelerate alpha-synuclein aggregation, because that's what we are looking at. If in the human intestine these proteins are there, probably that is where alpha-synuclein is getting aggregated. So we threw in some Hafnia CSGA into alpha-synuclein. As you can see, alpha-synuclein by itself. We added some Hafnia, and voila, we see an increase in aggregation of alpha-synuclein. And we did the same thing with Citrobacter. And interestingly, at the same concentration, Citrobacter did not actually accelerate alpha-synuclein aggregation, which was weird. We thought it would do the same thing, but it did not. Remember, Citrobacter is a fast aggregating CSG, whereas Hafnia is a slow aggregating CSGA. So we hypothesized maybe the intrinsic amyloid forming ability of CSGA governs its ability to accelerate alpha-synuclein aggregation. So in a simpler words, probably the slow aggregating CSGA proteins are better at accelerating alpha-synuclein aggregation than the fast aggregating CSGA proteins, because that's what our data showed us. So we went back and asked, what is it that makes a CSGA-like protein aggregate slower or faster? And that work was done in 2010, way back in my lab, wherein this is a primary sequence of E. coli CSGA. And in the sequence, you can see there are these aspartic and glycine residues which I've highlighted, and we call them gatekeeper residues. Why we call them gatekeeper residues? Because they inhibit the aggregation of E. coli CSG by itself. intrinsic ability of E. coli CAG to aggregate is inhibited by these glycine and aspartic acid residues. Why we know that? Because when you mutate these glycine and aspartic acid residues, you get a protein which we call CAG A star, because it aggregates very fast. And because we have mutated these residues which were inhibiting its aggregation. And we were like, yeah, maybe the this looks very similar to Citrobacter CAG, if you remember from a couple of slides ago. So we looked and asked, maybe there are gatekeeper residues lacking in Citrobacter CAGA, and Hafnia CAGA probably has those gatekeeper residues. And when we looked at the alignment, which I'm not showing here, definitely Citrobacter CAGA did not have these glycine and aspartic acid residues in them. And that is why it aggregates so fast. So what I did was, I mutated the non-gatekeeper residues in Citrobacter to gatekeeper residues. What I did was essentially, there were missing aspartic acid residues. So I mutated a couple of residues and changed it to aspartic acid. And then, as expected, we see a delayed aggregation of Citrobacter CAG, because now we have introduced these gatekeeper residues which inhibit the aggregation propensity or aggregation ability of Citrobacter CAG. Based on a hypothesis, now this Citrobacter CAG gatekeeper should now accelerate alpha-synuclein aggregation, because remember, I told you fast aggregating CAG is protein do not accelerate alpha-synuclein aggregation, whereas slow aggregating one do accelerate alpha-synuclein aggregation. So if our hypothesis is correct, probably Citrobacter CAG gatekeeper, which is a slow aggregating version of the Citrobacter, should accelerate alpha-synuclein aggregation. So we did the same assay again. We took alpha-synuclein by itself and added Citrobacter CAG gatekeeper, which is a slower version. And now we see accelerated alpha-synuclein aggregation. And just to give you a snapshot, this is what the wild type Citrobacter look. It did not accelerate alpha-synuclein aggregation to a greater extent, but the gatekeeper one did accelerate alpha-synuclein aggregation dramatically, which lends more support to a hypothesis and also tells us the importance of how the protein aggregation kinetics governs its ability to accelerate or interact with different human proteins. So based on this, we propose a mechanism wherein if you have a fast aggregating CSGA, what happens is the CSGA, which is a fast aggregating CSGA like Citrobacter, it starts from a random protein, forms these intermediates and immediately goes into a fiber. And the fibers are very stable. So they do not interact with alpha-synuclein and they do not do accelerate alpha-synuclein aggregation. Because during the process from going from a monomer to a fiber is very fast, as shown over here. So there's less time for alpha-synuclein to interact with these aggregates or these intermediates and accelerates fiber formation. Whereas in case of slow aggregating CAG variants, what we think is that the process from going from a monomer to a oligomer to a fiber is it's comparatively slower. So what happens in this case, probably there's the intermediate or the oligomers over here have more more stable than the oligomers over here, because these oligomers immediately go into fibers. Whereas in this case, the oligomers stay there for a longer time before they go into fibers. So in this case, if you throw in some alpha-synuclein, now that alpha-synuclein can interact with these oligomers and start aggregating itself. It's like providing a template for the monomeric alpha-synuclein to start aggregating rapidly with respect to slow aggregators. And many of the proposed therapeutics in this field are actually looking at slowing aggregation of proteins or stopping the aggregation of proteins. But looking at this data, it looks like probably that might not be a good therapeutic idea, because if you slow the aggregation of gut bacterial proteins which form aggregates, probably it might be counterproductive. It might actually help alpha-synuclein aggregation in the gut. So just to summarize, I'll wrap it up because I've not shown a lot of things. The human microbiome, the gut microbiome in human do harbor bacteria which encode amyloid-forming proteins like CAG in the gut. So these CSG in protein accelerate alpha-synuclein aggregation in vitro, at least we see. And we also have some mouse studies which looks like they do the same thing in mouse studies, which I've not shown over here. And there seems to be a correlation between how a CSG-like protein aggregates and how it affects alpha-synuclein active aggregation. So there seems to the faster aggregating CSG are not good at accelerating alpha-synuclein, whereas a slower aggregating CSG at accelerating alpha-synuclein aggregation. This is my last point. And it seems like oligomers are something which appears to be the species responsible for accelerating alpha-synuclein aggregation. And with that, I'd like to acknowledge my university, and this is my lab. This is my boss. He's fun to be with. He's always doing crazy stuff in our pictures, and all my lab members. Of course, the funding from NIH, my graduate school, and my department is MCDB. And with that, I'll be happy to take any questions if you have. And I'm going to stop sharing my screen if that's okay.
That's okay, sir. Thank you very much, sir. That was a very inspiring and very informative talk from your side. Am I audible?
Yeah,
yes,
I can hear you.
You are.
Okay. It is very important for this lecture was very important for us, for especially those students who want to become researchers, and they should get out of their comfort zone. And that was a very interesting thing. There are few questions from our Devra sir only, and I also had some questions, but I would like to go for Devra sir. Please ask your question.
Yes, thank you, Rohan, for allowing me to ask questions. Sujeet, you said something about sporadic PD condition, right?
Yeah.
And can you explain how in case the bacteria is present in the gut, how it can lead to certain sporadic situations? Because it is going to feed in the CSGA to possibly every neuron, I believe.
So there's the leaky gut is the reason we think what's happening. There are enteric nervous system has enteric neurons, and we have a collaborator in Philadelphia, Shala Tukel. She's a Turkish scientist, and she has actually shown that in the gut or the enteric nervous system, enteric neurons which line the intestine actually take up CSGA proteins from bacteria. They can engulf it, and that is where we think alpha-synuclein is coming in contact. So we don't think the aggregate alpha-synuclein aggregation actually starts in the gut. It just might be getting primed there. It's not that it's getting aggregated in the gut. It might be just getting primed, and this primed alpha-synuclein, which is starting to aggregate or maybe in a oligomeric form, then is migrated via the vagus nerve from the enteric nervous system to central nervous system, wherein it comes in contact with abundant of alpha-synuclein, and then it starts aggregation of that alpha-synuclein. That's where we think that is happening. And mouse models have also shown that, we have germ-free mouse, and we inject them with, or we infect them with CSGA producing bacteria, and those mouse models actually develop Parkinson's. If you delete the CSGA from those bacteria which are getting colonized in the mouse gut, they don't suffer from Parkinson's. So that was, we still don't know how it is going there. It's just something is happening there, which
can we establish some kind of a correlation between certain other peptides or proteins which are produced by the bacterium to make the blood-brain barrier more permeable, maybe? I don't know.
Probably because dysbiosis is actually associated with more barrier permeability. The blood-brain barrier is actually more permeable if there's dysbiosis, and dysbiosis is dysbiosis because we still don't know what is a normal microbiome. So and our diet, the way we that all feeds into the gut diversity, and if it's not diverse, then definitely there's a barrier permeability, and then we are making things easier to get into the brain compared to a healthy gut. And I don't know what a healthy gut is. We have to still yet to define that.
Very interestingly, it's something like that if you don't have a healthy gut, or you are infected with those organisms which possibly you work with, within a matter of say 2-3 minutes, 2-3 hours, the person is susceptible to develop those amyloid fibers, right? It's striking.
It's striking. It forms. They are so robust, these biofilms, to get rid of. You know better, so you worked with them. So it's so robust, and it's wonderful to study them. And it's like they form a very robust biofilm. But probably once getting infected, we have infected mouse with biofilm forming bacteria. The process from there to the brain to Parkinson's probably is a decade-long process. It's not that it's going to happen immediately, but we are just accelerating it by having those bacteria in our gut, compared to not having those bacteria in our gut.
That also possibly predicts how long these bacteria stay inside you, and what kind of a relationship they share with the host symbiote bacteria maybe, and then there could be a kind of competition, and then further the things might.
So these are opportunistic anaerobes, and they kind of, we have seen them, they kind of outcompete the normal microbiome we have. because they have these, not only are they biofilm forming, they also produce toxins which, like hemolysin. We studied hemolysin back, I remember, when we were in my master. So yeah, so they also produce those kind of toxins. So there's definitely not good to have them in the gut. Yeah.
And how do they affect the dopaminergic neurons in PD? These synucleins and
They deposit
CSGs?
in the no, we don't know. We did some brain biopsies. We never saw CSG in the brain.
Okay.
But alpha synuclein actually deposits in the dopamine producing neurons, and as accelerates.
It creates a barrier possibly for the. Yeah.
It aggregates in the Lewy body in the form of Lewy bodies in the dopamine producing neurons, and that leads to the death of those neurons because just a huge chunk of protein in the cytoplasm.
Okay. Yeah, over to you, Rohan. I'm done with it.
Sir, some questions are there on chat box. Shall we go for that?
Yeah.
Okay, sir.
Go ahead.
One of our student is asking, Bhagyashri Likhitkar, is there any possible therapeutic interventions that could be applied in this case? On that note, also, I want to know that what kind of, if the diet is able to modify this behavior of bacteria, which can save the person from this Parkinson disease or whatever the other diseases which are now are connected to the gut microbiome.
Great questions. To answer the first question, yes, we are actually working with a small startup in Boston area which makes small molecules, and these small molecules are targeted towards the bacterial protein, either to make it aggregate faster or not aggregate at all. So there are a couple of therapeutics. You can actually stop that aggregation of the bacterial protein, which causes acceleration of alpha synuclein aggregation. You can stop it, or you can make it so fast that it never gets a chance to accelerate alpha synuclein aggregation. So there are two ways of thinking about it, and we are working with some chemists from Sweden too, who are making those rationally designing small molecules based on 2-pyridones is a base parent molecule which is published, and then we are decorating them with different alkyl groups, aryl groups, and whatnot. I'm not a chemist, I don't know that, but they are doing that, and we are working on that. Yeah, and there are some promising results, and probably they are looking for patenting their molecule. Second question about a diet and how we can do that. To be honest, that would be a good question to dietitian. I'm not sure what kind of diet would help our gut or what is healthy for a gut. I for sure know I don't eat healthy, so probably my gut is not healthy at all. So yeah, if you find the answer, do tell me.
Sure. One more question from our audience: is there any genetic modification which can be implied to treat Parkinson disease?
Say that again. Is there any genetic?
Is there any genetic modification which can be implied to treat Parkinson disease?
If I understand the question, are there genetic mutations which cause Parkinson's disease, or are there genetic mutations which can be done to treat Parkinson's disease?
I believe that's the person want to ask.
I saw a smile on Anshul's face. Might be Anshul is interested in answering that.
Yeah, Anshul, go ahead, doctor. Yeah, jump in if you have something to say.
No, I was smiling for the fact, maybe it's the first question that was asked, that what sort of genetic mutations actually may be leading to Parkinson's, because I don't think, you don't want to expose yourself to mutation to treat from Parkinson's.
Yeah, exactly. That is what I was confused. Yeah, there are mutations which cause Parkinson's. There are mutations in the gene for alpha synuclein. It's called the SYNCA, if I'm not mistaken, and those kind of lead to Parkinson's disease. But gene therapy, if you're talking about, is there gene therapy? Can you do some? No, I'm not aware of that. Yeah.
Keeping the topic of this webinar in mind, I would like to ask this last question: that you worked very long in an industry and a well settled job. It might be very anxious thing to shift from a well-paid job to a PhD. How you did that, and how you cope up with all these kind of things?
Well, I was young and it didn't make sense. I was young and I made mistake. No, I'm just joking. I was just young. It was an instinctive decision. I was getting too comfortable with my job, and I hate comfort zones. it means that I'm not growing at all if I get in the comfort zone. So I was like, no, this is not what I want to do. And I'm young, I can take risks. And I funded myself essentially. I told you, my mom and dad never went to college. So I saved money during my job in Tata. And I said, okay, I'm going to use that money to apply for grad school and get here. And here it's funded, completely funded. Everything is taken care of. All I had to be honest, I had to take a loan to fly out here. It was 70,000 rupees. And I didn't have that money because I didn't save that much. So it was fun, but it's still worth it. And it is definitely, it's coming here completely different. It's a cultural shock to come when you're here. Everything is different, right? From your food, the people, everything is different. But you get adjusted. People in general are good, and they'll help you out everywhere. And the transition was made smooth by having a great boss right now and great friends, colleagues over here. So it's a big thing. It's, what ifs, kya hoga, what happens, what will happen if I go there. It's cool. All you need to do is, just here goes nothing. That's the attitude I went with. Let's do it, and we figure it out. That kind of thing. I don't know how helpful it was, but that's how I approached it.
What suggestion you will give to our young students who are pursuing research or who want to pursue research in America or in India, and especially in the time of this pandemic and the political and economical situation going on in America and India?
Wow, I feel like a host on those news channels. You have a political analysis.
Not because everything has been affected.
Yeah. You don't need to answer this, okay?
Oh, okay.
I'm just joking. It seems like I'm a big expert on all this, but no. I don't know about the political climate. I mean, those are constantly changing dynamic things. I'd rather not go into politics. But yeah, as a researcher, definitely, I mean, applying or continuing research is like, what I learned in my research was asking the right question the first time. I wasted 3 years asking the incorrect question. It took me 3 years of work to answer the correct question. And once I answered the correct question, it was all easy after that. So, before even going into research, asking correct questions is something which I learned out of my PhD. Problem solving is something I learned out of my PhD, asking the correct question and then solving that problem. For the researchers here, I will also think, maybe sometimes taking a step back and, looking at the broader picture and saying, is this a correct question to pursue? Is this the right question? That probably would help. And in pandemic, I think in pandemic, you can still do a lot. Probably this application cycle for PhDs or masters in US is gone, but you can still work on, if you're working from home, if the college is shut down, you can still work from home, build on your career. LinkedIn is something I'm really into. You do that, network with people, network with scientists you want to get in touch with. If you want to get in US, email them. They're generally good people. They'll reply. Some of them do not. That's fine. Email them, talk to them. Tell them about what you are interested in, what you are, or what you would want to do when you're in their labs or, in their research or in universities. And networking is something which you can do at home sitting right now during the pandemic. Secondly, building your core competency, like, something I do. I'm a consultant. Consulting is something we do it's a non-profit thing in the sense we help businesses around Michigan area and solve their problems, which is something different, completely different from my research, which I'm doing. So I'm actually working as a project manager. I lead a team of people. For example, we had a healthcare company startup which came to us and says, we have this product, we want to, commercialize it, help us with it. So we all got together. We all PhDs, no MBAs, nothing, no business people. We just sit together, we brainstorm, and that is what we do. And it's just online everything, and we do it. So that is other things you can develop to probably, for lack of a good word, market yourself as a good candidate when you're applying to PhDs or masters in US.
Thank you. Thank you once again for your wonderful talk. Now I request Ms. Rita Lakkakul, Assistant Professor, Biochemistry, to introduce our next speaker. Over to you, Rita ma'am.
Thank you, Rohan sir. Sir, I'm audible?
Yes, Rita, go ahead.
Very good evening to all. I, Rita Lakkakul, Assistant Professor, Department of Biochemistry and Biotechnology. I'm honored and take this privilege to introduce our next speaker, Dr. Ansul Lokdarshi. Sir is a plant molecular biochemist and has done his master's from Hislop College, Nagpur. Sir has completed his PhD in biochemistry and cellular and molecular biology from University of Tennessee, Knoxville. Dr. Ansul Lokdarshi at present is a postdoctoral research associate at University of Tennessee, Knoxville. Sir has several publications in peer-reviewed journals to his credit. His research area basically focuses on translational control mechanism and has keen interest in developing new technologies for plant phenotyping. He is enthusiastic in developing new teaching tools for customizing undergraduate student learning assessment. Dr. Anshul Lokdarshi will be delivering a talk on the topic regulation of translation in response to reactive oxygen by the protein kinase Gcn2. With this brief introduction, I welcome you, sir, to please take over the session.
Thank you, Rita ma'am, and for the nice introduction, and again, Raut sir, for organizing this event. Let me start sharing my screen. Let's see if so. Okay, so can everyone see my screen? Okay.
Yeah, good to go.
I'm just gonna minimize this. Give me just a second. Oops. Okay, that should do it. We turn on the laser pointer. Okay, so to keep in focus for all the early career scientists as the major audience, let me start out by asking a fundamental question. So why do we need plants? I mean, so, the fact that everything we eat comes directly or indirectly from plants, and pretty much all the nitrogen that humans and animals have in their body. So, your building blocks, genetic material, DNA having nitrogen, mRNA, and then you have protein, it all comes from plants. And that should be enough to suffice that, that plants are very important for sustenance of human and life form. And again, adding carbon dioxide fixation, production of oxygen that we, that we live on, essentially, again, asserts the fact that plants are very important for sustenance of our life, all human and animal life on the planet. Now, as plants, as we depend on plants, plant health and yield is dependent upon one of the most important energy generating processes on the planet, called as photosynthesis. And I'm sure everybody must be aware of this. Now, sadly, photosynthesis, as it seems, it's not very efficient. And the net output is around 5% of the leaf efficiency. So 100% light absorbed, what you get is just net 5% is essentially used for making energy. And the rest of it seems to get lost in successive reactions. Now, of the many hindering factors that essentially suppress the net photosynthetic output, one of the major abiotic stresses that hampers this is called as excess light stress. And in very simple terms, it's actually the light that is absorbed beyond the photosynthetic capacity of the leaf. So to put this in perspective, I like food. Sometimes when you go to some parties, you tend to eat more than you're supposed to, and your stomach actually hurts. Think about plants don't have the luxury. They're sessile, they're at one place, and all the sunlight that comes through, they just have to absorb it. And they also suffer from stomach ache, as you can say, and that's essentially, you can think that that's kind of an excess light stress, okay? So how do plant cells, at a cellular level, how do they deal with this? What type of signaling response happens in that, and what type of stress response signaling happens during the stress? So if you look at, if you plot all the, series of events, biochemical molecular events that takes place from when a plant is exposed from a low light to highlights, example, early morning all the way to the afternoon, now you're facing highlight. If you plot that on a time scale, 0 to 1000 minutes, something that becomes, very interesting is that transcriptional response, which is, regulation at the level of mRNA, takes around 100 minutes, while the ROS accumulation, reactive oxygen species accumulation, so again, as a nice secondary messenger, that takes, close to a minute. So it's a very fast, quick, secondary messenger response. And translational response, so ribosome loading, so the mRNAs that are actively getting translated into polypeptides, that happens within 10 minutes. So what this, asserts that regulation of translation or the conversion of mRNA from the protein, that actually serves as a rapid stress stress response switch for mediating responses towards excess light stress. So translation is highly regulated in all eukaryotes. And of the 3 steps of translation, that is initiation, elongation, and termination, translation initiation seems to be one of the primary steps for a lot of regulation. Now, most of the sensing and signaling in plant, it occurs by these 2 major kinases. And I'm sure everybody must be aware of the target of rapamycin or TOR kinase that is essentially correlated with plant growth and development, and sometimes also with stress like osmotic stress. And TOR kinase, again, via its downstream targets, again, tends to regulate translation initiation. The major bulk of, you can say, this core stress response is actually handled by another kinase, which is called as the general control of non-derepressible 2, which was originally found in yeast. In short, Jc1 kinase that actually takes input from all these different kind of stress conditions like wounding, pathogen attack, amino acid starvation, ultraviolet light, and so on and so forth. And what it does is that it phosphorylates its substrate, which is the alpha subunit of this translation initiation factor called EIF2. And once EIF2 alpha is phosphorylated, that leads to stop of translation initiation, and no more translation goes on, and that leads to downregulation of global translation. And the idea is to save energy because you are under stress. That's the same, that's the whole idea over here. So now, quite interestingly, unlike mammals, I'm not sure how to, okay, I'll leave that there. So quite interestingly, unlike mammals, which actually have these 4 different kinases, so PKR, HRI, PK, and Jc1 kinase that happen to phosphorylate at a conserved serine residue in the EIF2 alpha, plants only have Jc1 kinase to respond to all the different types of stress. And the idea is that once EIF2 alpha is phosphorylated, it acts as a competitive inhibitor for a EIF2B, which is actually a guanine nucleotide exchange factor. So what it does is that it exchanges GDP to GDP and charges the EIF2 alpha for another round of translation initiation. So if you don't have this EIF2 GTP, you're going to block the translation initiation, and that's how this whole module actually functions. So Jc1 phosphorylates EIF2 alpha, and once phosphorylated, you block the translation initiation. And like I said, all plants have just Jc1 kinase that phosphorylates EIF2 alpha. So it's quite interesting that it actually gives another incentive of using plant Jc1 EIF2 alpha module as a nice model system to dissect the function of Jc1 kinase, and that you can apply also in animals where if EIF2 alpha is phosphorylated, it will kind of convolute the results because you don't know which kinase actually phosphorylated in case of some particular stress. So we can use this nice model system. So as I said, Jc1 EIF2 alpha module is highly conserved across all eukaryotes, and the most interesting part is that all plants have Jc1 kinase to phosphorylate EIF2 alpha. Okay, so one of the classic ways, how you look for Jc1 kinase activity in any model system is by performing immunoblots, or as we say, Western blots, and we use a phosphor-specific EIF2 alpha antibody. And shown over here is an example in Arabidopsis thaliana. So the top part over here is a phosphoimmunoblot showing phosphorylated form of the EIF2 alpha. So that's the at time 0 background phosphorylation. And within 4, 6, and 12 hours, you can see there's an increase in the amount of phosphorylation signal that is detected. This is a proxy for the increase in Jc1 kinase activity. Okay. And this is in response to an herbicide, which is chlorosulfuron, which targets branch chain amino acid metabolism that happens in the chloroplast. Tubulin over here is used as a loading control. So Jc1 also responds, like I said earlier, to different types of stress. Over here, ultraviolet radiation leads to Jc1 activation. Again, Jc1 dependent EIF2 alpha phosphorylation in wild type seedlings. And once you have the GCN2 knockout, that means there is no GCN2 protein being made in these seedlings. You don't see any phosphorylation, again signifying that, okay, it's actually GCN2 dependent phenomenon. Again, GCN2 kinase is activated by defense hormones like salicylic acid. You can see that, there is nice activation, excuse me, methyl jasmonate, and the ethylene precursor ACC. So again, involved in also, taking a lot of input from different types of stress conditions. So we can say that GCN2 phosphorylates AF2 alpha in response to wide range of abiotic stress conditions. So let's look at some of the observations here. So we know that excess light is actually a form of, it's a type of photooxidative stress that lowers the photosynthetic output. And the regulation of translation, so the level of mRNA to protein synthesis actually serves as a rapid stress response switch for, again, a rapid adjustment towards high light stress. So we want something like that. We want, this is a dynamic abiotic stress, and we want something that is very rapid, to kind of respond to this dynamic environment. And GCN2 AF2 alpha module is a core stress response component of the translational control in plants. So based on these observations, in today's talk, I'll be addressing these three main questions. So is GCN2 involved in mediating responses towards excess light stress? Given the fact that GCN2 AF2 alpha module is again a core stress response, so maybe it does mediate excess light stress. How does GCN2 how does the loss of GCN2, so what I mean by that, how do the GCN2 knockout mutants, they behave under excess light stress treatment? Because there won't be any phosphorylation of that. And do stress conditions that actually mimic this whole, the mechanism of excess light stress, which I'll be showing you in just a couple of slides. If they are able to mimic this whole same pathway, can they actually activate the GCN2 kinase? So just to look at, to answer this question is that, well, we first need to understand, does this excess light stress actually lead to formation of reactive oxygen species in plants? Okay, so here is the classic experimental pipeline. So we grow Arabidopsis thaliana seedlings on again plant media, and they grow under 16-hour light, 8-hour dark period, which is again classic long day conditions, and we subject them to 24-hour dark acclimation. And the idea over here is that, whatever reactive oxygen species are getting produced in the plants, hopefully 24-hour dark acclimation will again, subdue it, take it to the lowest level. And at the end of 24-hour dark acclimation, we name this as a time point 0. Okay. And to induce or challenge the seedlings with excess light stress, we re-expose them back to the same light intensity over here, 80 microinstein light, and this is called as the excess light stress treatment. Okay, so we collect a seedling sample from excess light stress treated seedlings, and then we collect sample from T0, that is right at the end of 24-hour dark acclimation. And shown over here is confocal images of 24-hour dark adapted seedlings. You can see that, you can hardly see any ROS fluorescent reactive oxygen species, which is actually detected by this fluorescent dye that reacts with the reactive oxygen species, and it produces fluorescence in the GFP, green fluorescent protein wavelength. Okay, now, but once you give the excess light stress for 30 minutes, you do see this patchy, patchy appearance of ROS fluorescence. And if you zoom in closely, you can find that this fluorescence seems to appear mostly from the chloroplast. So chloroplast, you can see these round structures, chlorophyll has its own red autofluorescence. And since this ROS fluorescence dye appears to be green, the overlap is sort of a yellow color. So most of this yellow fluorescence seems to come from specifically from chloroplast. So this is something that has been shown earlier in studies saying that, pretty much 90% of the reactive oxygen species during the daytime, during the light period, is actually contributed by chloroplast. Rest of it comes from other organelles. So We know that, in our experimental setup, we can reproduce pretty much this system that excess light stress triggers ROS accumulation specifically in the chloroplast, and ROS accumulation seems to be in a localized pattern, not all over the leaf, but there are areas, patchy networks where, you can see the ROS accumulation. So next thing is that in order to test that, well, can we activate GCN2 kinase by using the same setup where we actually trigger excess light stress? Okay, so in order to do that, we do the same setup where you have 16-hour light, 8-hour dark grown seedlings. We subject them to 24-hour dark and we re-expose them back to the light. Okay, and so give it excess light stress. We collect a sample at 24-hour dark, which is right over here. That is T0, and a red arrow means right after this the excess light treatment has been given. So what you see over here is that this top part is the immunoblot for with the EIF2 alpha, phospho-specific EIF2 alpha antibody, and you see that within 30 minutes there is a nice EIF2 alpha phosphorylation signal. Again, it shows that GCN2 is now active. Where at time 0, which was 24-hour dark acclimation, you don't see any GCN2 kinase activity. Okay. Now, this is a GCN2 dependent phenomenon because, again, in GCN2 knockout seedlings where you don't have any GCN2 protein, you don't see any phosphorylation at all. Okay, this middle panel over here is Poncio S staining, which shows equal loading in all the lanes, and again EIF2 alpha levels, which is just the total protein, does not change along this time course in the wild type as well as GCN2. Okay, now this excess light stress triggered GCN2 activation is also dependent upon how much light you are going to apply. Okay, so 2 microeinstein, 25 microeinstein, 80 and 200 microeinstein. You can see that at 25 microeinstein, it does induce GCN2 activation, albeit at a lower level compared with what 80 microeinstein does. And it seems that by 80 microeinstein, you are seen to be saturating the amount of GCN2 activation. Okay, so what you can conclude is that EIF2 alpha is phosphorylated in a GCN2 dependent manner under excess light stress, and GCN2 activation follows light dosage. Okay, so we can see kind of a model developing over here is that excess light stress triggers chloroplastic reactive oxygen species that seems to activate the cytosolic GCN2 kinase, which phosphorylates the EIF2 alpha. Okay, well, how can we test that this reactive oxygen species from the chloroplast is indeed coming from the photosynthesis? So it's actually the photosynthetic ROS that is leaching out or somehow activating the cytosolic GCN2 kinase. So in order to test that, we use 3 types of photosynthetic inhibitors or herbicides, you can say. One of them is called as DCMU, which essentially blocks the electron transport between quinone A and quinone B. So this is again, the classic Z scheme of photosynthesis. I'm sure everybody must be aware. DCMU tends to block the electron transport between these quinones, and what it leads, the end result is extremely low amount of reactive oxygen species, which are singlet oxygen, hydrogen peroxide, or the superoxide. Okay, so keep in mind DCMU blocks reactive oxygen species. We use another photosynthetic inhibitor DBMIB, which blocks the function of cytochrome B6F complex, and the end result is that there is indeed production of superoxide, which gets converted to hydrogen peroxide, but it's a very slow process. So you're going to delay it, and you will have some production, but it's a delayed effect. On the other hand, we use methyl valogen, which is also called as paraquat, which is a classic herbicide used in agriculture. And this is an extremely potent herbicide which generates massive amount of reactive oxygen species, especially it generates superoxide that can get converted to hydrogen peroxide. So again, the idea is that we can use DCMU, block ROS, DBMIB, delay the ROS, MV, create a lot of ROS. So let's see what happens to the GCN2 activation. So as expected, mock treatment. So again, the idea is that we grow seedlings 16-hour light, 8-hour dark, we give 24-hour dark acclimation, and then we give excess light stress. So you can see that GCN2 is activated. But before giving excess light stress, we can actually add these DCMU. You can see you can block the production of ROS, because as I said earlier, and you will suppress GCN2 activation. You can apply DBMIB, which tends to delay the ROS production, and you have delayed the activation of GCN2. Unlike, you can see 30 minutes, that's pretty much nothing. It's only at 2 hours that GCN2 gets active. On the retrospect, methylvologen, which is a very potent herbicide, you can see it pretty much nukes the whole blood in a way that it has hyperactivated GCN2 kinase within 10 minutes. Again, this suggests that the hydrogen peroxide majorly, which is generated in the chloroplast under excess light stress, results in GCN2 mediated EAF2 alpha phosphorylation. So, well, as I said earlier, there are other organelles. You can question that mitochondria, endoplasmic reticulum, they do generate reactive oxygen species. So the question is that how specific is this GCN2 kinase activation by chloroplastic ROS? So to test that, what we did was, we grew the plants again, seedlings in 16-hour light, 8-hour dark, and we subjected them to 24-hour dark acclimation. And here's the trick that, we did all our experiments under dark itself, to avoid any light mediated, again, in a chloroplast involvement reactive oxygen species. So these experiments have been done after 24-hour dark acclimation in dark, okay? So to induce mitochondrial reactive oxygen species, a classic way in the literature is that you give hypoxia, which is low oxygen stress. So in general terms, flooding stress, and reoxygenation, which is after hypoxia has happened, that tends to generate superoxide in mitochondria. And sham and antimycin A, they tend to block their mitochondrial electron transport chain inhibitors. And once you add them, these all chemicals and the reoxygenation process tends to generate mitochondrial reactive oxygen species. Now, neither of these case, these reagents were able to induce any GCN2 activation. Same thing with tunicamycin, which is a classic endoplasmic reticulum stress inducer. It triggers reactive oxygen species in the ER, also does not induce any GCN2 activation. So this is quite interesting to know that GCN2 activation signal is indeed specific to the chloroplastic reactive oxygen species. Now, so we know that excess light stress triggers chloroplast reactive oxygen species, and that somehow is activating a GCN2 kinase in the cytoplasm that's phosphorylating EAF2 alpha, and there must be something going on at the level of translation. Well, if you remove GCN2 kinase out of the equation, which is GCN2 kinase, now how are the plants going to respond to highlight stress? What kind of the phenotype are they going to show under excess light stress, since they may have some mismanagement of energy conservation? So in order to do that, we perform this highlight stress sensitivity assay. And the idea is that we have 3 days old wild type Landsberg plants, and we have GCN2 knockout mutants. Like I said earlier, there is no GCN2 protein here. And we have a complementation line, which is we put back the GCN2 gene back into the GCN2 knockout mutant. So it's, most like, you can assume it's like a wild type, okay? And the idea is that to give a continuous light as a control and continuous highlight. So we are talking about 80 to around 800, okay? So 10-fold more light. And you can see that, after 3 days of continuous highlight treatment, we measure primary root length as a diagnostic. It's just a diagnostic test for looking at plant health, okay? So you see that, the primary root length of GCN2 mutants is lower compared with the wild type. And once you put the gene back, the primary root length is restored, showing that there is indeed a function of GCN2 in maintaining plant health. And this ties up to this, whole idea about excess light stress, chloroplastic ROS, and there is an important function of this GCN2 dependent EAF2 alpha phosphorylation. There is another diagnostic test we do to look at plant health, especially GCN2 health, and that is by measuring the fresh weight. We just take these seedlings out and we measure the fresh weight, and you can see that same GCN2 seedlings are significantly lower. The fresh weight is significantly lower compared to the wild type and also the complementation line. So this asserts the idea that loss of GCN2 increases sensitivity towards highlight. Okay. Now, yes, excess light stress triggers chloroplastic ROS, and this model is starting to become clear. Now, are there other stress conditions that can actually induce production of reactive oxygen species in the chloroplast and activate GCN2 kinase? So the question is that, do other stress conditions that mimic the excess light stress by upregulating chloroplastic reactive oxygen species activate the GCN2 kinase? So we have recently, we published this, we have shown that cold stress Again, 4 degrees, you can take the plants, grown at 16-hour light, 8-hour dark at 22 degrees and shift them to cold stress, that is 4 degrees. And you can see that, there is GCN2 dependent upregulation of eIF2 alpha phosphorylation. So there is an exacerbation. You can see there is an increase within 10 minutes of GCN2 activity. ZT2 is at at 22 degrees, you give 10 minutes of 4 degrees, there is an increase in phosphorylation. Salt stretch, which is NaCl, also increases GCN2 activation within 30 minutes and 2 hours. There's, you can see, a very robust phosphorylation. Mannitol is a sign of osmotic stress, used as a control over here. It does not induce GCN2 activation compared with salt stress. Now, we wanted to test, plant pathogen. Again, this is all abiotic stress. What about biotic stress? So the link is methyl jasmonate and salicylic acid, which are defense hormones. We also show that methyl jasmonate and salicylic acid, they require light, to activate GCN2 kinase. So methyl jasmonate, once you treat it after 24-hour dark acclimation, you can see hardly any activation of GCN2 kinase. While once you apply the same hormones in light, you can see that, GCN2 kinase is rapidly activated within 10 minutes. Okay, so this again, the whole idea is that you can start applying these stress conditions, but you need light because all these stress conditions somehow are feeding through chloroplastic reactive oxygen species to activate the GCN2 kinase. So to summarize this model, where this GCN2 kinase, this whole model stands right now is that different abiotic and biotic stress conditions like salt, cold, excess light, even day-night cycle where you have light intensity increasing during the day. So circadian or a dial cycle, you can say that ultraviolet light, behind the shade is herbicide, salicylic acid, biotic, defense hormone, jasmonic acid, and we have also, we are testing these bacterial pathogens like Pseudomonas syringae. All these different types of stress conditions, they somehow are causing this fine balance between ROS production and its quenching in the chloroplast to be thrown off. The equilibrium gets thrown off, and what you start making is a lot of reactive oxygen species in the chloroplast, and somehow this reactive oxygen species is shifting the equilibrium and activating the GCN2 kinase. Now, interestingly, GCN2 kinase functions as a homodimer, and it's actually associated with the 40S ribosome. So we are talking about now, regulation of translation. So it's sitting on the 40S ribosome, and you have the translation initiation factor eIF2 alpha, eIF2 and alpha subunit with a serine 51 residue here. And under no stress condition, you have the mRNA, which is now, this is the 48S pre-initiation complex. Everything is fine. Next 60S comes in. Translation is a go, okay? But once you have a stress, reactive oxygen species somehow activate the kinase, and it phosphorylates this, serine 51 of the alpha subunit of eIF2. And what it does is that it kind of locks this translation initiation complex with the mRNA in its own state, and it inhibits this, no more translation going on, okay? So the mRNA just stays there. So what we are looking is, some of the questions we are trying to ask now is that how does chloroplastic ROS actually activate this kinase? How can, this ROS like physically go and activate the GCN2 kinase? What kind of biochemistry, is involved in this? What is the nature of the GCN2 regulated mRNAs under stress? So we are trying to, do what is called as ribosome footprinting. So we are trying to sequence these mRNAs that are in these ribosomes by deep sequencing and understand, what's the nature. We want to identify these mRNAs, and is there some similarity between the stress condition, or what kind of specificity does it have? And since I said earlier, GCN2 kinase, all plants have GCN2 kinase and eIF2 alpha. So does this model actually apply in crops? Because eventually we really want to take this into and understand if we can overexpress GCN2 kinase, or we can underexpress it, how can we help crops create better resilient, and how can we increase the plant health and yield of the crops? So with this, I would like to thank my PI, Albert von Arnim. He has been extremely instrumental in this study and motivation. The lab member is Ricardo. He is the resident bioinformatician graduate student in our lab. And again, I didn't show, we have a lot of sequencing, RNA-seq and translatomics done. He has been extremely helpful in that. Our collaborator, Dr. Bradley from Michigan State, he has been involved with the plant pathogen side. Funding is from National Science Foundation. introduction. And again, University of Tennessee BCME department has been helpful in everything aspect, and especially, thanks to our undergraduates. So I had the luxury, I have to say, that having such phenomenal undergrads like Phil, Michelle, Madison, and Zoe, some of them first generation undergrads, I should say now graduates, and they have been co-authors in our 2 recent publications in Plant Cell and Frontiers, and they are, without them, this project was not possible. So they have been amazing undergraduates. Okay, and again, this is my Twitter handle if you want to get connected, Lokdarshi_Ansul. Okay, so before I go and say, I'll happy to answer questions, since Devraj sir had pointed out just like a 1-minute summary, where I was and what I, where I am and what I mean to do, plan to do next is that, again, I'll sort of echo what Sujeet had already said earlier, is that, so I did my bachelor's and master's in biotechnology from Hislop School of Biotechnology, or now Department of Biotechnology, Hislop College, and I had I'd taken botany at that time. The idea was that, okay, India is an agricultural land, I'll have good career prospects in botany, and I did, to be honest, I was very lucky to have these amazing instructors, or I would say mentors. Devraj sir, Dr. Avinash Upadhyay, Mashitanam, the phenomenal people who have given tremendous guidance and support. So and I did, again, did specialization in plant tissue culture, a lot of techniques there, and brainstorming, and so the one of the message that I have to say that over here is that you really need to identify a problem. That will help to understand, there are many ways to solve it, but you need to identify the problem, and who you work for and what you work is central to success. That combination, if you find like who you're working for and what you work is going to help you clear your vision. Once I did my bachelor's and master's, again, the same path. You give GRE and TOEFL, you apply to, reach out to professors, different universities. Again, they're very kind. It's not a scary thing. Make sure your cover letter is really good. And you reach out to folks, just say, hey, I'm interested in your research. Make sure you read about the research, and you reach out, and there is a response. You will get a response. And then the idea was that, again, I did the same thing. I came to UT Knoxville. It's closer to the Oak Ridge National Lab. Again, I did my PhD in biochemistry here. Now I'm a postdoc at the same place. And again, the take-home messages from this part of the tenure during my PhD and postdoc is that, keep in mind that, we don't plan to fail. We always fail to plan. So make sure you when you start out PhD, or when you're coming before even that, make sure you have a plan that what you want to do next. Okay. And we often tend to have this imposter syndrome that, okay, we did bachelor's, master's, here's my PhD, and academia is the only route. Tomorrow I'm going to become assistant professor, and that's how. No, you have to branch out. Like what Sujeet is saying. Again, a lot of people are going to say the same thing, is that you can go to industry, you can do many things, scientific writing. There are so many careers. So as soon as you're able to realize, about your imposter syndrome, it's going to help you a lot. And I've been actively involved in, these organizations, international organizations like American Society of Plant Biologists, ASPB. There is a section, Early Career Plant Sciences section. I'll really, encourage everyone there to go and check it out. And you can follow ASPB, ASPS. There is a lot of things to learn about how early career plant scientists in United States, or all around the world, what they're doing. So just open up the mind. And last thing I have to say is that, later on at some point, I've discussed this with Devraj sir, is that we are going to prepare we are trying to prepare something like a go-to handbook for, early career scientists, that something like us, how we started, not only from the stage of when you are in undergraduate, or BSc or MSc, all the career and the personal and professional guidance that you can get all along the PhD and even postdoc. We're trying to prepare something, a handbook like this, and hopefully we'll have out sometime soon. Okay, so that's all I have.
Thank you, sir. Thank you. That was indeed a splendid talk, sir. Dr. Devrat Begre sir has few questions for you.
Rita,
Sir, over to you.
are there no questions from the chat box?
No, sir. There are no questions from the chat.
Okay. Okay, Anshul, I have a few. Pardon my knowledge in plant bio or chemistry or biotechnology. Okay, I am not a plant biochemist or biotechnologist. And two, had I been good enough, then possibly you would have landed up something doing other than plants, maybe, because I am not a plant biotechnologist.
So having, having said that, since you brought it up, remember this system is a very conserved system that is actually found in animals also. So, the value of this system is using plant model system. You can, study that and you can apply to any other model system, C. elegans, fly, or a human system. So that's
right. I was coming to flies because my interest is in flies. See, the concept is, I don't know, but I have been trying this since years, that in case we expose the flies for a prolonged light phase or light period, then they tend to age early. So can there be a correlation between the reactive oxygen species generation and GCN and fly aging maybe?
Sure. Yeah. So again, like I said, in Drosophila or like in a fly system, there are the other three kinases other than GCN2. So PKR,
right.
PERK, and HRI. So now, having said that, you're right. Dial cycle has been shown. This day-night cycle does control translation. It's very, in all model systems, it's known, and GCN2 kinase does play a role in that. So, how does that fit in? You're right. Maybe again, exposure of these flies to abrupt changes in light conditions, example, continuous light, maybe, affecting translation, and that might be going through GCN2 kinase, which is again in plants. It has, our work shows that. So, yeah, that's possible.
And something you can shed light upon for your light-induced pathway.
Yes.
Is there a protein that you can track which possibly senses ROS production and then carries it to GCN kinase, so that there is a pathway that you can correlate? Only because, just a question, just because you were talking only about chloroplast originating ROS.
Yeah, so that's actually not a naive question. That's the $1,000,000 question. What activates the GCN2 kinase from the chloroplast? And that's, we know the agent over here is a reactive oxygen species, but you're right. It could be ROS that leaks out from the chloroplast and has some protein modification on the GCN2 kinase. One, or it could be, you're right. Maybe some other protein that is actually acting as a mediator, you can say that, that is getting activated by our ROS and then activate. So it could be a direct as well as an indirect effect, and we don't know right now. Yeah.
And did you try exposing your cells to exogenous ROS?
Yes. So what we did was, to actually test this before even excess light stress, we wanted to test that, if you apply the bad, neat, and quick way of doing ROS experiments for plants, as we do, just add a bunch of ROS, which plants don't get that all the time. We actually did take Arabidopsis seedlings, and we gave a 24-hour dark acclimation, and you spray them with hydrogen peroxide, and you can actually hyperactivate within 10 minutes in dark itself. So application of just directly hydrogen peroxide does activate GCN2 kinase, and that actually led us to the next experiment, like I said, with the excess light stress.
Right, and the final thing is, like, we have found this out, that the methionine stress in Drosophila leads to a kind of premature aging in Drosophila, and there has been several papers regarding this, and this has been correlated with ROS disbalance. Rather, I will not call it stress, because redox signaling plays a quite important role in terms of several pathways. You can the most important one is differentiation pathway. So just to have a kind of, you can say, a comment, did you try methionine stress?
So I haven't tried any methionine stress per se, but to get back on that, GCN2 activation, I didn't go into the details, is that so GCN2, the activation pathway of GCN2, which is primarily based on yeast and human studies, is that it has a C-terminal histidyl tRNA binding domain. So what that domain does is that when you have, say, amino acid stress, okay, you're not consuming, cells are not getting enough amino acids, your tRNAs that you're going to have, they don't have amino acids anymore, right? So they are called as uncharged tRNA. Histidyl tRNA domain that is present in the GCN2 can actually bind to it, and it pops open, and now the kinase is active. Okay, so GCN2 kinase is a sensor for amino acid stress in the plant. Okay, so one way in plants, how is this test has been done, is actually apply herbicides. So chlorosulfuron, glyphosate, farmers use Basta or glyphosate, which has been in use all the time, is that these classic herbicides, they actually target amino acid metabolism in the chloroplast. And we have shown in our studies is that if you add chlorosulfuron or glyphosate, you can actually activate GCN2 kinase. So that goes back to your question, is that if you cause any amino acid stress, in the plants, it also activates GCN2 kinase. But you haven't tried methionine or specific amino acid like that, and just to add to that, all these herbicides that people have applied and used this in past, they also need light to activate GCN2 kinase, because you can apply herbicides in dark, you won't activate GCN2 kinase. So chloroplast ROS is very important.
And does this anyway boils down to sulfur content of the herbicides as well?
Yes. So that's an excellent question. There is a study that came out that links the TOR kinase and the GCN2 kinase, and there is a sulfur or cysteine, how sulfur metabolism or cysteine metabolism is involved. And I'm sure, everybody, like you are aware, and everybody must be knowing, again, it goes back to ROS, antioxidant and metabolism, because again, cysteine is
ultimately it leads to that only.
Diagnostic for ROS. Yeah. I can see there's a question coming from Utpal here, that did you study the ROS scavenging system, or antioxidant system? Yes. So we have done treatments with antioxidants before you expose plants to excess light stress. Okay, and we do see that antioxidants, again, ROS scavenging systems, they actually suppress the GCN2 activation specific to chloroplast ROS. Yes, we have done those studies.
So there is one question from the participant. Can we take it?
Yes.
From Bhagyashri, she's asking, if we use C4 plants in the experiment, what possible changes we would have to make in the light exposure cycle?
That's an excellent question. Okay, so one of the ways, so to answer that question again, 24-hour dark acclimation, that hour of acclimation has to be optimized. Let's put it that way. Okay, so so you can actually assess for EIF2 alpha phosphorylation, which is a diagnostic for GCN2 activity, and you'll have to optimize what number of hours that you do a dark acclimation to the C4 plants that will lead to the basal level of activity or disappearance of activity, and that's how you have to pan out your experiments. Yeah.
Thank you, sir. I once again thank you for being with us. I now request Mr. Pradeep Hirapure sir to please continue this session.
Thank you, Rita madam. Good evening, everyone. I am Pradeep Hirapure, Assistant Professor, Department of Biochemistry and Biotechnology, Dr. Ambedkar College, Diksha Bhumi, Nagpur. I am very happy to introduce our next guest speaker, Dr. Sunayana Mitra. Dr. Sunayana Mitra did her master degree in biotechnology from Nagpur University, Nagpur in 2007. She has completed her PhD in biochemistry from Laushania State University, Baton Rouge in 2015. Currently, Madam is working as a postdoctoral research fellow at Department of Biomedical Engineering at Michigan State University, Michigan. Madam has many awards and several publications to her credits. So we are very lucky to have with us such a renowned speaker for this session. Now I welcome to Dr. Sunayana Madam to deliver her expert talk on the topic novel magnetosensing protein. Please, Madam, take over the session. Thank you.
Thank you, sir, for such a nice introduction. So I will quickly start sharing my screen. All right. Okay. Thank you for inviting me, and thank you to David sir and the organizing committee. My name is Sunayana, and I am a postdoctoral fellow at the Institute of Quantitative Health and Health Sciences and Engineering, or the IQ Institute, as we like to call it, at Michigan State University, the other public university in Michigan. We are in East Lansing, USA. So the title of my talk is Shining a Light on a Novel Magnetosensing Protein. So a little bit of background. I'm going to talk about the protein which was recently discovered by our lab. It's called electromagnetic perceptive gene protein, or EPG in short. So various aquatic species and even land species in the Earth use Earth's magnetic field for orientation, navigation, detection of prey and predators. And one of these is the glass catfish, or Cryptopterus bicerus. And you can see this picture actually doesn't do much justice to it. It's called glass catfish because it's completely transparent and you can actually see its all organs. And it has a particular protein which we call the EPG protein. And this was discovered by my lab, Prof. Asaf Gilad and Dr. Galit Pellard lab in collaboration. And so this protein is basically the bread and butter of our lab. So we study it. And what this protein does is it responds to electromagnetic stimuli, which causes changes in the intracellular calcium levels. So why do we care about this protein really? So before that, I want to show you a good quick video. These are the glass catfish in an aquarium. And we are going to introduce electromagnetic stimulus from the right. And so you will hear a noise, and that is the start of the magnetic stimulus. And the fish actually swim away from the magnetic stimulus together once you turn it on. So this is how the fish responds to electromagnetic field. So why do we care about the EPG? Because our lab is a basically synthetic biology lab. We do a lot of molecular biology, and we think that there is a lot of potential of EPG as a therapeutic candidate. It can give you non-invasive control of cellular activity. So what we want to do is create biological circuits. So maybe use EPG as a gene therapeutic agent and use its ability to release calcium on electromagnetic stimulus to drive maybe another gene of interest, which may be a disease-causing gene. And we have seen that it is cell and location specific. And so the other lab that was involved in the discovery of EPG is a pure neuroscience lab. So we are interested in developing gene therapy agents for diseases like epilepsy and Parkinson's, etc. Maybe sort of drive these, maybe identify certain therapeutic gene targets and then use EPG to control by calcium influx. So what do we know about this EPG protein as so far? We don't know much. We don't know the structure of the protein, but we can make some educated guesses depending on the function. So it has a 20 amino acid signal peptide region. So this particular section of the EPG protein sequence is probably a signal sequence, and we think it's a membrane signal. And then it also has a particular domain, which is called the Lys6 antigen or urokinase gen type plasminogen activator receptor like domain. I'm not going to go into what that means, but basically it is similar to a cell surface receptor. And then we also think that there is a possible transmembrane region at the N-terminal from how it anchors onto the membrane. So that is basically what we schematic of what we think the EPG protein looks like. So then the other thing that we know, that when we did immunohistochemistry on the EPG protein, and here we can see pan-cadherin is a membrane marker, cell membrane marker, and the EPG is in red. And if you can look here, you can see that the EPG co-localizes with the pan-cadherin at the membrane, and it indeed tells us that EPG is membrane bound in cells. These were HEK cells. And then the other thing we know, that when we transfect neuronal cell culture cells with EPG, there is increased in calcium indicator dye. So what it does is, what this Furet dye does, it changes the dye intensity changes with the level of calcium present in the cell. So once this panel shows the EPG expression, and then this panel here shows what happens at baseline, where there is no electromagnetic stimulus at the Furet levels. And then when we stimulate it with a static electromagnet, there is a significant increase in EPG, or rather significant increase in calcium levels, or which can be seen by this increase in Furet levels. So we want to know more about this cell, more about this protein. We want to harness it. We want to use it to create gene therapy agents. So next, what we want to do is, what we set out to do is look at whether this EPG protein actually expresses in other cell cultures. And then what I did was took hex cells, and then we transfected it with this particular EPG construct, where which EPG is, there is a TD tomato linker here with, and then, so basically there is a internal ribosomal entry site between EPG and TD tomato. So if EPG is expressed, the TD tomato would also be expressed. And so we did see a lot of expression of the TD tomato being in red. We saw a lot of expression of EPG. And then we also did Western blot comparison between EPG and hex cells transfected with EPG and hex cells without transfection. And what we see is generally EPG likes to dimerize, and we generally see a 26 kilodalton protein. Maybe you can see there is a very faint band of the 13 kilodaltons, which is the monomeric EPG. So next we wanted to, we can express it and we can see it, it is working fine. So what if we could see this EPG protein in real time under magnetic stimulation? And what if we could see what it does when you actually actually stimulate it with a magnet, and you can actually follow the protein activity? So for doing that, we need to use a technique called single molecular imaging using total internal reflection fluorescence microscopy. This technique is very interesting. It was developed at the University of Michigan in early 1980s. And what it does is it allows the imaging of fluorescent molecules close to the glass water or glass specimen interface. So if you look here at this panel, at the schematic here, we can actually see, instead of a normal fluorescence microscope where the entire cell is excited with light and then you capture the emitted light, the fluorescence, we actually you actually look at the reflection of this critical angle, and which illuminates only a little bit of the cell which is between the slide and your cell, and you can see the membrane. So it's like 100 micron field of depth, and this allows for nice observation of membrane-associated proteins. You can look at cell adhesion, hormone binding, molecular transport, endo and exocytic processes such as neurotransmitter release and uptake. So then, since we know that it's a membrane-bound protein and we want to follow it real time, this looked like a good technique to use. And we used a halo tag, something called halo tag imaging. It's a fusion, you create a fusion protein with your protein of interest, and this halo tag protein binds covalently with a ligand dye, and then this is a very how to say it? It's a very robust binding, and then it can actually use to direct imaging of protein trafficking, protein turnover. You can label live cells or fixed cells, and you can also do purification if you need to. So with that, we wanted to test it first and look at it under a normal fluorescence microscope, see everything is working. And so I'm not going to go into the details, but I created a fusion protein with halo tag, and then I labeled the cells with either Alexa Flor dye or Genelia Flor, and I'll explain a little more. And then when we looked at epifluorescent microscopy, this is what I saw. You don't need to adjust your screen. I actually didn't see anything. So that was when I thought, okay, what's going on? Why I can't see anything? And then I looked at, this was with Alexa Flor. And when I looked at Genelia Flor, I actually saw some cells. So it was not that my experiment didn't work, but there was something going on. So this is a schematic of the EPG molecule with the halo tag fusion protein. And if you can see, this is where we think the halo tag goes at the C terminal, and this is the putative transmembrane region. So since Alexa 488 is a cell impermeable dye, the only way we would be able to see Alexa 488 if it was this orientation at the bottom, where the halo tag is on the outside and the EPG is on the inside of the cell, but we don't see it. So it's possible that EPG is a membrane-anchored extracellular protein. So, but we didn't need to look at it a little bit more. In case of Genelia Flor, since it's a cell-permeable dye, either of those two orientations is possible. So to investigate further, we tried a different strategy where we cloned the halo tag on the N-terminal of EPG. And what happened this time is, yes, we could see the Genelia Flor because the Genelia Flor is cell-permeable. So again, either of these orientations would work. But with respect to Alexa Flor, now we can see the same dye where we had earlier not seen anything. Now we can see this nice expression of the dye. And that is because here also we can see that Alexa Flor can work only in one orientation of EPG, where the cell EPG protein is anchored by its membrane, anchored by its N-terminal, and I'm sorry, anchored by its C-terminal, and is an extracellular dye. So that led us to conclude that the EPG protein is a membrane-anchored extracellular protein. And now we move on to the actual turf microscopy. So what I did here is I took HeLa cells and I transfected them with the EPG construct, and then I imaged them using the total internal reflection fluorescence microscopy. And then I tried to look at the same cell before any magnetic stimulation. I imaged them with magnetic stimulation, and then I removed the magnet and then wait for 5 minutes and then look at the stimulation, look at the magnetic response again. So these individual dots that you can see are the actual EPG molecules that we can track in real time. So let's look at the video. So the first is before there is any magnetic induction, and then there is with magnet, with the magnet right there, and then we have the after magnetic induction. So what does it tell us here? So, sorry, I'll move on. Okay, so we have this hypothesis now that there is a possible EPG mechanism that is going on here, where we have some EPGs on the cell membrane, and then we expose it to magnet. And what happens is it causes recruitment of EPG from inside the cell to outside. And it also causes maybe dimerization and multimerization of cell. Maybe it also interacts with other proteins in the cell, and that leads to increase in calcium ion influx inside the cell. So that is basically what we think is our basic mechanism. So what we went on to do is, let's look at this particular phenomena and see if we can find a way to look at it in details. So we did something called a pulse chase experiment. What we did was we transfected the cells with the same type of EPG halotype DNA, and then we added a pulse of the Alexa dye that will basically label the entire surface pool, and then we added different levels of the Janelia flow dye that would lead to labeling only the internal pool of the EPG molecule, because that's the Janelia flow dye is basically the cell permeable dye. Now, what is why we did that is basically because the EPG dye, the EPG molecule that we saw in the previous video, there's too much on the surface, and that amount of protein is not easy to quantify, or we cannot know the behavior of this protein. So that was the reason to label the entire surface pool. And this is what happens. So when we labeled with Janelia floor, so these were imaged simultaneously with both channels together. And again, the similar with and without magnet. You can see the Alexa floor dye similar, there is similar amount of dye of Alexa floor, but there is an increase. Again, these single red molecules that are running around on the cell surface are EPG molecules. And our goal is now this is a work in progress, and our goal is now to quantify this particular protein, maybe quantify the dots. Is there increase in number of EPG molecules? Is there clustering? Is there a change in any kind of physical property? So what we are going to do now is do something called particle localization and tracking. And this, what it does is we have a cell, and then a cell, we have particles which we are tracking individually. And when these particles are on the cell, they are very dynamic. They move around and follow a certain path, which is called the track or trajectory. And they jump from one point to another, and these are called jumps. The number of the jumps and the length of the trajectory can give us important information about this particular protein. So what this MATLAB program does is basically it took quantum dot labeled protein, and then it can give you a 3D map of the particles shown as a peak. And it develops an algorithm where you can look at the trajectory of the protein, look at where it's whether it's stopping, whether it's moving faster or slower. Let's say a protein is forming a monomer or dimer on stimulation, then the protein will become slower. If more protein is coming out from inside the cell to the membrane, we'll see a different way of movement. And then these individual particle location and trajectories are generally used to detect particle diffusion coefficients. And that is something that gives you an idea of what the protein is doing on the cell. Is it slowing down? Is it moving faster? Is there a change in stimulation with electromagnetic stimulation? Is there a change in protein behavior? And so that is where we are at right now. And this is where we are going to look at. So now that we know the EPG protein is membrane bound and there is some response to magnetic fields, that we want to do more research into it. So in summary, EPG is a magnetosensing gene with high therapeutic potential, and it is a membrane anchored extracellular protein. The magnetic stimulation probably causes the recruitment of protein to the membrane and causes clustering. We are doing single particle tracking and analysis to analyze the behavior and properties. So I would take any questions. I would just want to thank everybody. This is my lab. This is the person in the center is Dr. Asaf Gilad, my PI. Wonderful person to work with. This picture was taken almost a year back. We haven't taken new pictures yet, but our lab has shrunk quite a bit because of lack of summer students, because of obvious reasons. I would also like to thank Dr. Galit Pellet from Pellet Lab and Vijay Krishnan, who have been instrumental in discovering APG, and also would like to thank the lab of Ian Schmidt and Carlo, who have the microscopy instrumentation. And that is basically all that I have for now. I did miss the memo about talking about my journey from Hislop from Nagpur University to here at Michigan State University. So I want to put a few minutes of that. I am actually from Calcutta and I moved to Nagpur for my studies. I did my bachelor's from LAD College and then I did my master's from Hislop School of Biotechnology as it was known then. And I had some wonderful mentors both at my bachelor's and my master's, including Devrat sir, and everybody inspired me a lot. My initial goal was to do a PhD, whether in India or outside, it didn't matter. But the funny thing is I took the NET exam and then I was able to clear the first part. I don't know if it's in 2 parts still. I was able to clear the first part, but I wasn't able to clear the second part of the NET exam. But in the meantime, I was doing GRE as well, preparing for it. And I managed to clear GRE and TOEFL. And here I applied to several universities. I'm trying to get my... I applied to several universities and was fortunate enough to get into one. My graduate journey has not been easy, I would say, because the original lab I joined in was, I had a very nice mentor and professor, but 3 years into the program, he had to leave the lab and I had to, after 3 years into my program, I had to move into another lab and start all over again. But I finally managed to complete my PhD in 2015. And then I moved to Baltimore to do my first postdoctoral project. And what happened is, even though I really loved the work and it was something very different, we were looking at noise-induced hearing loss. My husband and his lab decided to move here. And so I thought, okay, maybe this is a good chance to move over, try something new. And so I moved here too. And I had volunteered in Asaf's lab before, right after my PhD, and he was kind enough to take me. And so I would like to say that there have been a lot of ups and downs in my scientific career. So my advice would be keep your doors open and never lose hope, and always, there will always, if one door closes, some door will open. And then if you have any questions, concerns about your career activity or planning, there's my email right there. I would try to help you any way I can. And I would take any questions. Thank you.
Thank you, madam, for your excellent presentation and throwing lights on such a novel topic, and which is very important for us. Ma'am, your presentation will be definitely helpful for all the participants to think in new direction research. Now the session is open for discussion.
Pradeep sir, if you can allow me, I have a few questions.
Okay, sure. You can ask.
Sunayana, that was quite an interesting topic that you are dealing with. Just a few questions in my mind. Are there any reports of this magnetosensing protein expression in brain? I would like to.
If I mean the only report, only paper that has been published so far is the original paper, and they have actually done. They are actually now doing some injections where they are trying to do EPG expression in the rat brain. So it's in the work. In the works, yes, you do see expression of the EPG in the brain. You see fluorescence. We don't tag the EPG directly, but it's like an indirect tag where you put internal ribosomal binding site, and so you see fluorescence, and you know that the protein is being expressed. Yes.
Since neurons are quite sensitive to calcium ion concentration,
Yes.
then possibly we can externally induce those neurons and can lead to certain calcium ion concentration change inside the cell, and perhaps can what you can say influence neuron firing. Then that's
Right,
why I
right.
was
That is
quite
the whole
interested.
plan. Yeah.
Right. Okay. And because it's still a naive topic, will there be any effect of say MRI in on the function of brain in case such proteins are expressed in brains and they are documented to do that?
That is a very good question. Actually, the whole idea is to what if we could use MRI as a therapeutic agent, as something that we could have. I mean, there is a huge, there's going to be a huge body of work needed from going from cell culture to an actual person who would have this kind of a gene therapeutic agent which responds to migraine. But it would be nice to be able to use something like MRI, which we already know that is already approved, and use it in a therapeutic way and not just imaging. You can actually do therapy as well as imaging while you are doing therapy, something like a functional MRI. So yeah.
And maybe correlating what your research work is and what Anshul was talking about, then there could be certain external physical agents by the way of which without going for any intervention we can do some treatments. Then maybe in near future you can lead up into that.
Right, right. The only, we want to do it non-invasively, but you still have to introduce the gene as a gene therapy agent in some way. So that would be invasive. But yes, that is always there is always a way to do that.
Right, right.
Hi, Sunan. Here. I have a question.
Sure.
Okay.
Do you think the membrane anchoring of this protein is necessary for its activity? Can you get rid of that membrane anchor domain? Or, since if you're looking at the therapeutic aspect, probably you have to make this protein functional without being tethered to a membrane or getting rid of its membrane anchoring.
That is a good question. I've been working on that. So this has been a side project actually, but my original project, being a biochemist, was to try to crystallize this protein. And when you remove the membrane anchoring region, it actually refuses to express for some reason. I don't know if it's maybe because it's in bacteria or something, but haven't had much luck seeing even expression of the EPG properly. Maybe it helps in folding or something, but yeah, you're right. But we can't even see it in the cell anymore if you remove the membrane anchor.
I see.
Yeah, maybe because it must be associated with in some or the other way with the calcium ion, right? Calcium ion channel, right?
Right. That
And
is the
that has to be membrane associated in order to show the influence of it.
that is true. The thing is, the membrane bound region is so small that we're still trying to figure out if there is a role of it anyway. There is a tiny 10 amino acid intracellular region of this protein, which is like a bit of it sticking out into the cell, and that is too short for it to associate it with other proteins, but maybe there is a role. we still don't know a lot about this.
Once again, thank you, madam, for your wonderful session. Now
Thank
I
you.
request to Ms. Swati Chimburkar, madam, to continue next session. Thank you.
Thank you, sir. I hope I am audible to everyone.
Yes, madam.
Okay, sir. Good evening,
Yes,
everyone.
Swati, go ahead.
I, Swati Chimurkar, Assistant Professor, Department of Biochemistry and Biotechnology, Dr. Ambedkar College, feels privileged to introduce today's speaker, Dr. Depanwita Pal. Ma'am has completed her master's from Nagpur University in Biochemistry stream and completed her PhD with distinction from University of North Texas Health Science Center, Fort Worth. Ma'am is presently working as a postdoctoral research associate in Shriners Hospital for Children, Portland, and also worked as research specialist and graduate teaching assistant. Ma'am has number of publications to her credit with research experience in different projects and has been honored and rewarded for young research, new investigator recognition, as well as short talk training award. With this brief introduction, I request ma'am to address the gathering. Over to you, ma'am.
Thank you, Swati. Thank you for that introduction. Can everyone hear me okay?
Yes, ma'am.
Okay.
Yeah, Depanwita.
All right. All right. So I'd like to start by thanking the organizers, especially Utpal, for this opportunity to talk to students. It's always fun, and especially since I was a student not very long ago, it's even more. Okay, so I work in an orthopedic hospital, and so our area of research is pertaining to musculoskeletal tissues. So mostly muscles, bones, and tendons. And our lab focuses on tendon development. So I'll be telling you a little bit about that. But before I go into that, let me tell you how did I land here. So like Swati said, I studied in Nagpur, and Devrat sir just told me that Alka ma'am is on this call, and now I feel absolutely nervous because she was my first teacher of biochemistry. So I did my BSc in LAD College, and during that time, I was lucky enough to intern for a little bit at NERI and CICR, and then moved to the University Department of Biochemistry to do my MSc. And then from there, I moved to Fort Worth to do my PhD, which was also in biochemistry and molecular biology. However, having been trained as a biochemist and a molecular biologist, I now actually work in developmental biology, which is completely different. So I had to learn a lot of new things. So this is to tell you that it doesn't matter what you're trained as. Science is very diverse, and your training will help you in no matter which field that you go. Okay, so coming back to what I study now. So like I said, our lab focuses on tendons. So let's start with what are tendons? So tendons are these dense connective tissues that connect your muscle to the bone, all right? And they basically transmit the force of muscle contraction to the skeleton. So any joint movement that you do, if you're typing, if you're eating, you're playing cricket, any sort of joint movement requires a coordination between the muscle, the tendon, and the bone. And if you see here, so this is just images of your leg when you're walking or running, you'll see that the tendon will basically elongate or recoil based on the molecular cues that it gets from the muscle to the bone. So it is absolutely central to any sort of joint function. So if you have ever felt pain after you've been playing, or if you feel pain in your wrist because you've been typing so much, I can assure you it is your tendon. So why are we studying this? So even though I told you tendons are extremely important and they're central to any sort of joint function, there is very, very little known about them. Compared to muscles and bones, we know absolutely nothing about how tendons grow. So it's a very under-researched tissue. However, it has extensive clinical relevance. So just to give you some numbers, if you look in the world, musculoskeletal disorders range from about 14 to 42%. And in India, that number ranges from 20 to as high as 90%, which is very high, in fact. So, and 50% of these musculoskeletal injuries actually involve your tendon. So basically, we are trying to treat a tissue that we know very little about. When you go to a doctor for pain and it turns out to be your tendon, there are not many things that they can tell you to fix it. They talk about cortisol injections, they talk about physical therapy. At some point, they talk about surgery. But again, none of these treatments are very effective. And part of the reason is that tendon heals extremely poorly. And one of the reasons for that is we still do not understand how tendons grow. And this is actually a major roadblock in any sort of clinical progress. So this is the gap that our lab is trying to fill. We are trying to understand what drives tendon growth. If I want to grow tendon in a lab, what can I use to make it grow? If you have a tendon injury, say you have a torn tendon in your knee or your Achilles is torn, what can I do to make it better? So that's where we are focused. So what do we know about tendons so far? So our lab has previously shown that there are two major steps for tendon growth. So the first is induction. So induction basically means what is the first appearance of tendon cells in your body. So we work on mice. So I'm going to be talking about E days. So E basically stands for embryonic day, okay? So in mice, you start seeing tendons. So the blue and the purple that you see here around embryonic day 12. So that's the first appearance of tendon cells. So these are the earliest cells of a tendon. So we call them progenitors. And then after that, what happens is basically as your bone and skeleton grows, your tendons basically elongate to accommodate for the skeletal growth, okay? So basically think of it, so tendons are elastic structure, you know, they can be stretched. So basically what's happening is as your bone is growing, your tendon is already connected to the muscle and the bone, and it's basically just elongating to allow for additional growth. So despite years of research, we have only 3 transcription factors that have to date been associated with tendon cell fate. There are scleraxis, mohawk, and EGR1 and 2. These are the only 3 factors that we know so far that have any sort of role in tendon development. And I know I cannot belabor this point enough. It's a very, very small field. If you have only 3 factors that are associated with a particular cell specification, that's a very low number. And our lab has in fact shown that out of these molecules, scleraxis is probably the only one that has a very definitive role to play in tendon development. So that's what I'll be focusing on for the rest of my talk. So here I'm going to show you why we have focused on scleraxis. So scleraxis is a very characteristic marker for tendon cells from the very early stages throughout development. So here I'm basically showing you, these are mouse paws. So think of it as, you know, mouse hands. This is starting from embryonic day 11. So right now it's just a bud. There are no indentations. As you keep going forward, you begin to see these fingers form. And the purple that you see here, this is basically RNA expression for scleraxis. So you see from embryonic day 11, as you move forward in time, you begin to see these tendons form. And scleraxis basically labels these tendons very well. It is in fact the only marker that labels tendon cells from these early stages, and it stays throughout development. So it's a very powerful marker to study tendons. And our lab hence subsequently generated a transgenic tendon reporter mouse. So what that means is we basically replaced part of the coding region of scleraxis gene and added a fluorescent protein there. So this is the GFP sequence, and we have just added it into scleraxis. So every time there is scleraxis in any animal, you'll basically, if you look under the fluorescence microscope, your tendons will glow green. So here, this is again a mouse paw. And if you can see these numbers and all the green that you can see in this image, these are all your tendons. In fact, if you were to look at any of your tendons, any of your hand tendons under a microscope, this is what you would see. This is the kind of tendons even we have. So this became a very powerful tool for us to actually study tendons in real time. Okay, and the way we use this mouse is that this is in the background of all our studies. This is our basic study model. And what we can do is, if we section this tissue, gradually, we can actually get in much more detail what happens in your hand or even your leg. So like here, if you cut through at the finger level, at the digit level, this is what you get. So here again, all the green that you see are all tendons, and I have used an antibody to stain for the muscles. So in your digits, you do not have any muscles. You do not have any muscles in your fingers. Okay, so you're only looking at the tendons here. Then as you move a little forward, this is around your palm region. Now you begin to see, so this is around here. You now begin to see these muscles and the tendons. And then again, this is in the wrist region. You have more muscles there. And again, the size of your hand, you can see it's, increasing. And then again, in the arm, you can look at the muscles and tendons here. So basically, we use this to study how the tendon is growing. So if we have certain molecules that we think are important for tendon growth, or if we are interested in certain genes that we think might have a role in tendon development, we can actually mutate that gene or delete that gene, and then again, ask what happens to this tendon structure here. So our most important question, the question in our lab has always been, scleraxis has such a good expression in tendons, so obviously it has to do something, right? So to answer that question, we basically deleted scleraxis and we made a scleraxis mutant. So to do that, we took the mouse scleraxis gene and basically removed exon 1. So once you remove exon 1, now this mouse does not have a functional scleraxis gene, okay? So I will try to play this video here. Okay, so if you see, this mouse is actually, it doesn't walk on its paws, okay? It walks on its wrist because it doesn't have tendons in its hand. So even though this mouse is viable, it has mobility issues and it cannot move its tail. So it has a loss of a lot of tendons that I will show you in a minute. Also, if you put it back, if you lay it on its back, it cannot turn around, because for that process, you need to use your tail, and this mouse is unable to do that. So if you look closely at the tendon structure, so this, I'm showing you a regular mouse, you can see that there are multiple tendons in the hand, okay? This is if you're looking from the top. If you look in the mutant, again, here, you can see that there are a few tendons, but the most of the long tendons are actually lost. You do not have these big tendons that actually allow you to do this motion or this motion. And then again, if you look on the palmar side, now you have these tendons here in your palm, which are again missing in the mutant. So this mutant has very, very severe loss of tendons, suggesting that indeed scleraxis is very important for tendon growth. And in fact, when you look closely, what we realize is that the phenotype is a failure of tendon elongation. So I told you before that there are two major steps for tendon growth. The first is induction, and the second is elongation. So here I'm showing you sections through the knee. So this is your knee bone. Okay, so all the green that you see are bones, the red that you see is muscle, and the purple that you see is scleraxis expression, so that's tendon, okay? So this is your knee bone. These are the muscles of your thigh, and there are these tendons that actually connect your knee bone to these muscles, okay? It allows for your knee movement. But if you look in the mutant, now what you see is there is some tendon there, but it has failed to grow. It has failed to elongate. So basically, when you lose scleraxis, you're losing the ability of these tendons to elongate. And previous work from our lab discussed how are cells added to a growing tendon. What is elongation of tendon? So elongation is basically addition. You're basically adding cells to the tendon. So these can either be cells that are proliferating from within the tendon, or you can have cells that are being added from outside. So outside of the tendon, there might be cells that are actually getting recruited to the tendon. And in fact, we have shown before that this elongation is a combination of both proliferation and recruitment. And in fact, if you look in the hand, that becomes even more obvious. So here we are looking at just this region. And these, the green that you see here again, those are tendons, and we are looking at somewhere about here, okay? So what you see is that between embryonic day 13 and embryonic day 15, you have a fully formed tendon, okay? You have all the tendons that you will need. And the length of the tendon almost increases 1.6-fold. But if you look at within the tendon, here we are using BRDU, EDU, and Ki67. All of these are markers for proliferation. You'll find that there is less than 2% proliferation. You do not see any cells that are labeled between these tendons. If you look at the graph here, again, similar thing. What I've done here is I've sectioned this limb up to this part, and then I've counted the number of cells that are proliferating. So if you look at the number of cells that are proliferating, again, it's about 1%, okay? But if you look at the tendon length between these two stages, it's almost double of, you know, at E13 versus E15, it's almost double. So the tendon is obviously growing. And if it is not proliferating, that means it's almost exclusively dependent on recruiting new cells from the outside, right? So that actually showed us that recruitment is a very important aspect of this elongation. So we asked what would happen to recruitment if we deleted scleraxis. So what we did here is we used a Cree-Loxp system. And basically what you can do is you can label cells using a Cree. So these are cells, we used a different kind of Cree to label all the cells within a tendon. And then we used that Cree to delete scleraxis. So when you cross these two mouse, what you get is a deletion of scleraxis in those cells. So here, again, here we are looking at tail tendons. So the enlarged images here are at this point. And I've basically divided the panel into the 3 colors or 2 colors and a merge. So here we know that all the cells are labeled with the scleraxis GFP. So we are using the scleraxis GFP mouse. So all of the cells are labeled green. And then we have used a Cree and we are using red to denote the Cree. So again, all the cells are also red, okay? So when you look under the microscope, they'll actually look yellow. So both the green cells and the red cells here, they are both positive for scleraxis, right? This is a regular mouse. Now what we do is, now we use the Cree-Loxp system to delete scleraxis. So now you're deleting scleraxis only in the red cells. So your green cells are still intact, okay? So the very first thing you will notice is the size of these tendons. They're very, very small. And if you look at the red cells, they have big holes in them, okay? So when you look at a merge, now you can see that the green cells make the tendon. They are much smaller in size. And the red cells are basically just surrounding the tendon, okay? They are not able to enter the tendon. They're not getting recruited. So basically, the red cells do not have any scleraxis. So what this experiment shows is that scleraxis is absolutely essential. The expression of scleraxis is absolutely essential for the cell to identify that it needs to get recruited to the tendon. So if you do not have any scleraxis, all these cells that are getting incorporated into your tendon will not be able to, all right? So our next question was, what if, so these mice did not have scleraxis to begin with, you know, even before the tendons start forming, there is already, you've removed scleraxis from the equation. So now what if we could start the growth of the tendon, and then delete scleraxis. So you already have, recruitment has already started, and now you delete scleraxis from that equation. So for that, we used a slightly different system. It's also a Cree system, but this Cree gives you temporal control. So what I mean by that is you get to define at what time point you want to delete your gene. So what this Cree has is a ERT2. So that's basically a variant of an estrogen receptor. And if you give tamoxifen to this mouse, then you get an active Cree and you get deletion. If you do not give tamoxifen, there is no binding to the Cree, and so you do not get an active Cree. Okay. So again, we are using the same model here. We are using a flox scleraxis mouse. And then we are using a different version of Cree. So what Rosa Cree does is it labels all the cells in your body, not just tendons, everything. Everything is labeled. Okay. And so all the cells in the body will actually, once you give tamoxifen, will not have any scleraxis. So what happens there? So we saw before that between embryonic day 13 and embryonic day 14, there's a huge growth in these tendons, right? So we decided that we want to delete scleraxis in this time point. So again, I injected tamoxifen at E12 and E13, and then you let the mouse grow, okay? So you let the mouse grow up to E16 and E18, and then we look at the tendons at these two different time points. So here I'm showing you the tendons of here, your hand, right about this region. So this is one tendon and this is another tendon, okay? So these are your regular mice, and you can see you have nice big tendons that are actually functional, that look functional. But if you look at the mutant, you can see this big gap of region where you should have got these cells. Or even if you compare the size, they are much smaller in size as compared to your wild type. So basically what this shows is that not only scleraxis is important for the cell to get added to these tendons, but even if you already have a tendon that has started growing and then you remove scleraxis from the equation, the tendons do not form well. So the limb tendons have a continued dependence on scleraxis even after the growth of the tendon has already started. So what I've shown you so far is that scleraxis is a very prototypic marker for tendon cells. It's in fact the only marker that is present in the earliest progenitors of tendons. I've shown you that when you lose scleraxis, it leads to tendon elongation. So the tendon does not elongate to accommodate for your skeletal growth, okay? And that leads to severe tendon loss. This tendon elongation is a combination of addition of cells either by proliferation or recruitment of new cells. And in fact, the hand is very much dependent on this recruitment of cells. And for this recruitment of cells to happen, you need scleraxis expression. So the recruitment is dependent on scleraxis expression. And interestingly enough, even after you have started the growth of tendons, they have already formed, even then, if you lose scleraxis, you will not get a fully formed and functional set of tendons, okay? So naturally, we find that this aspect of scleraxis function is very heavily tied to recruitment, okay? And recruitment means that the tendon is recruiting these cells from outside of the tendon. And that was very curious to us. What are these cells that are outside the tendon that are actually, getting added to the tendon? So our question was, what are these recruited cells? And this is one of our ongoing studies. So what are these recruited cells? So the strategy that we are using to identify these cells is using a combination of scleraxis positive and scleraxis negative cells. So we know that if you have scleraxis, there will be recruitment. If you do not have scleraxis, there will not be any recruitment, right? So we can use this model to actually ask, what are these recruited cells? So the newest, biggest boom in our field right now are next generation sequencing approaches, okay? This is basically sequencing of your RNA or your DNA or epigenomics, so different kinds of sequencing approaches at single cell level. So we are using one of these next generation sequencing techniques to basically compare our scleraxis positive mouse with our scleraxis negative mouse. And what that should tell us is we know that these cells cannot get recruited. So if we can look at the genetic signature for the negative mouse and compare it, we might be able to tell what is the genetic signature of recruited cells. So the technique that we are using is single cell RNA sequencing, and it means just that. Basically, what this allows you to do is sequence your tissue of interest. So in our case, we sequence our tendons, and it allows you to sequence the RNA within every cell of the tendon. So if there are, say, 10,000 cells in your tendon, we can sequence each and every one of those cells to look at the genetic code of each of these cells, right? So this is a very powerful technique, and it is used in a lot of fields. It's being used in cancer, it is being used in Alzheimer's, it's used for a lot of different questions, especially because you can answer a lot of different topics with this one technique. You can look into tissue characterization. So you can look at, your tissue is made up of different cells, but you can identify each of those different cells using this technique. Then you can also see cell transition characterization. So, you start with, for example, a blood cell. When you start, the first blood cell is not a specialized blood cell, but eventually it becomes a specialized cell. Or even an immune cell, it doesn't start as a specialized cell, but over a course of time, it'll actually transition to become a specialized cell. So using this technique, you can actually look at the genetic signatures, and you can trace from the earliest cell to what happens to that cell, okay? So our experimental approach is, again, we take mouse limbs from both scleraxis positive and scleraxis negative. So basically you see these tendons, we basically isolate these tendons, dissociate them. So we can use different enzymes to basically dissociate them into single cell suspension, okay? And after we have gotten the single cell suspension, we basically do RNA-seq. So what that means is we isolate the RNA, code it into the DNA, and then look at differential signatures for every single cell in that tissue. So for this particular experiment, we used upwards of 8,000 cells for each of these groups. And the kind of data that you get back from this technique is looks like this. So it's a bit of a math magic that always makes me think that I should have paid more attention to math when I was in school. But nonetheless, so if you look at this graph, basically every dot on this graph is a cell, okay? Every dot on this graph comes from one of these samples. And if you look at, say, this red cluster here, all the cells that make up this red cluster they probably have the same genetic signature. So they are similar kind of cells, and that is why they have clustered together in the same color. Now, if you look at this green cluster right next to the red, what that means is that even though these green cells have a very slightly different signature compared to the red cells, they are connected. So think of it like, if this is you, this is your first cousin, this is your second cousin, this is your very, very distant cousin, and this guy is not from your family, okay? So this kind of a graph basically explains that. So not only does it match each cell to its signature, but it also tells you if two cells are connected in any way. And when I say connected in any way, it's genetically if they are connected, okay? So after we get this kind of a graph, and if you get, when you look at this kind of a signature, you can actually go through each of these clusters and look at what kind of genes make up those clusters. So there are typically upwards of hundreds of genes in each of these clusters. I'm showing you just the top ones. And when you look at these genes, we are able to identify them as, okay, this is a very, very tendon cluster. this is probably contamination because, there are muscle cells in here. This particular cluster has a lot of proliferating cells. So when you look at the signature, you're actually able to identify what kind of signaling molecules made up that cluster, what kind of signaling molecules allowed the growth of that kind of cell. So that's very powerful information, especially if you're trying to identify a very small subset of your cell population. The other thing that you can do is also compare. So we started out by asking what is the genetic makeup of a recruited cell? Can we use a genetic sequence? Can we use a gene sequence to say what kind of cells will be recruited to our tendon? So for that, we wanted to compare the scleraxis positive with scleraxis negative cells. So when you compare these two cells, these are the genes and these are the pathways that are upregulated in your scleraxis positive cells, okay? So we can actually look at these genes here as candidate molecules for what scleraxis does. So scleraxis, like I told you, it's a transcription factor, right? So that means it will either allow for a gene to get transcribed or it will block a gene from getting transcribed, right? So there has to be something that is downstream of scleraxis that mediates its function. So these candidates give us a good place to start looking for what could be downstream of scleraxis, what actually mediates the function of scleraxis. So instead of a needle in a haystack, we have needles in a haystack that we can look at, but it massively narrows down our list of genes that we want to look at. So this is where we are right now. This is what I'm doing for this project right now. So for future directions, like I said, we are trying to identify gene signatures for recruited cells and validate them if they are a potential candidate. And today I told you just about one aspect of scleraxis function. However, we have also shown, and we have data to show that scleraxis plays multiple roles. The role that I talked about is, embryonic role, but once the tendons have grown, once the mice are born, scleraxis also plays a role in tendon development at that stage. So I'm currently also doing some mutant studies to answer those questions. And if you remember, I started by telling you that there are only 3 transcription factors that are associated with tendon differentiation. And we now know that, so even if you delete scleraxis, you're still getting tendons. You are losing 90% of your tendons, but you're still getting 10% of your tendons. So we are still missing a gene that will delete, all the tendons. Can we get to a point where we can explain which tendon is formed thanks to which gene? So to do that, we are also part of this huge transcriptomics project, where again, we are using single-cell RNA-seq to possibly identify additional regulators of tendon sulfate. And with that, I would like to thank my lab, especially my mentor, that's Renan, and our lab manager, Brian. So Renan and Brian here. And Alice, she was a former member of our lab. She now has her own lab. She did some of the very definitive work early on in the scleraxis project. In fact, she started this project in the lab. And then we have computational biologists who help us with the math that I told you about. And then the cores that allow us to do these fancy techniques. And our funding comes from, for this project, comes from Shriners Hospital. And with that, I'd like to thank all of you for your attention. I'll be happy to answer any questions if you may have. So thank you.
Thank you so much, ma'am. That was really wonderful and informative session and interesting too.
Swati, if you may allow me, I have a few questions to ask.
Sure, please.
Okay, Depanwita,
yes,
not
sir.
my field, but quite an interesting topic. I have a few questions. If I have gathered it correctly, what you were trying to say, the scleraxis protein has been shown to be expressing very early in the embryonic development, right?
Yes.
Have you tried to find out that whether this transcription factor is expressed elsewhere also, other than in tendons, other cells?
Yes. So interestingly, these scleraxis is also expressed in ligaments. So like I said, tendons connect muscle to the bone, ligaments connect bone to bone, right? So it's also expressed in ligaments. Unfortunately, similar to tendons, we know absolutely nothing about ligaments also. So that's another good way for us to look at this. Other than tendons and ligaments, it's also expressed in hair cells. So if you look at, when you look at mouse, on their nose, they have a few hair follicles. And those hair follicles actually also express tendon. And then you have it in testes and in your heart. So heart is made up of these fibroblasts. And scleraxis has been shown to be an important regulator of these cardiac fibroblasts. Yeah.
I was about to come to that because you said that scleraxis is involved in recruitment of the cells, maybe from different origin.
Right.
There could be something interesting about finding out that what's the level of scleraxis expression in testis germinal stem cells. Maybe that can pave some way for you to understand that what exactly it is doing there.
Yes. So one of the questions that we have been trying to answer is, what kind of cells make up a tendon? And now that we know that there are cells that are coming from outside, that's one of the reasons why we are using single cell approach, because we have no way to directly label these cells. So perhaps this could be an indirect way to kind of get to that question. Yeah.
And perhaps, meaning I have been interested in germ cell differentiation as well as tracking certain factors which are involved in triggering differentiation of germ cells. See, it's fascinating to say like in the germ cells continue to give rise to their differentiated products all throughout the life of an individual, especially in case of testes. And if you can correlate it with possibly scleraxis expression, I'm not sure, but maybe there could be some correlation between reactive oxygen species or redox signaling and possibly scleraxis expression. I don't know. I'm just,
that's actually a very good.
Because
that's a very interesting.
Because redox signaling plays a quite important role in case of germ cell differentiation, especially in case of testes. And to get it even further, there has been a correlation that has been established between expression of pi RNAs, which are associated with transposon regulation, and germ cell differentiation. And all this, maybe in future you can correlate and find out.
Yeah, so interestingly enough, I'm also the tendon transcriptomics project that I told you about. That's one of the other major projects that I work on.
Yeah.
And one of the early hits that we saw, we got a lot of hits that we were expecting, but one of the early genes that we saw in there were mitochondrial genes. And we were very curious as to why we are seeing mitochondrial genes in tendons. So it could have something to do with the reactive oxygen species. Yeah, we haven't explored it as much yet, but that's something we are hoping to do. Yeah.
And any piRNA signatures in your transcriptomics data that you were, or DeepSeq data that you were gathering?
I'm not sure if the way that we are scanning the data, I'm not sure that we will see piRNA signatures. We would need to add an additional algorithm to the data. But that's very interesting because especially not piRNAs, but link RNAs have been shown to be very important for, in general, musculoskeletal growth. So it's, and nobody has actually looked at piRNAs with respect to musculoskeletal tissues. So that's a good point. We haven't looked, and it would be an interesting place to start. Yeah.
And is there any correlation that has been established with your protein? I regularly get it out of my mind, right? So I'm not naming it. Scleraxis, right? I got
yes.
It. Okay. Correlation of your protein expression with respect to where I was going to. I just got lost. Okay, if I get it, I will ping you. No
Sure. Sure. Thank you, sir.
So, ma'am, we have a question from one of our participants. What
Sure.
happens if expression of sclerexis is blocked after a tendon is fully formed?
That's a very good question. And I have spent the entire of last year answering it. So like I said, we used the pre-ERT2 system to basically delete scleraxis in newborn tendons. So in mice, you already have full tendon formation by embryonic day 14. That would compare to, I think, 40 days in the human embryo. So you already have a fully formed tendon structure. What happens after birth is addition of matrix. So like I said, these are dense connective tissues, so it has a lot of collagen in there, right? So if you look at all the skin creams in the market that say, oh, we can make you look younger, what these skin creams actually have is collagen. That's the gene in there. So what we find is when we delete scleraxis after the tendons are fully formed, is it does not affect your tendon structure. So you get the tendon structure, but it affects the matrix. The collagen that is in there, that is what gets affected. So even though it will not delay your mobility, you will still have full mobility, but your tendons will be very weak. They will not be as strong. And we are trying to do some of those mechanical studies right now to actually ask if what kind of weakness, like if I were to run, would that affect it more? If I were to walk, would my tendon tear? So those are some kind of the biomechanical questions that we are trying to answer now. Yeah, that's a very good question.
Okay, thank you.
Depanwita, I have
yes, sir.
It struck my mind.
Yes.
Sclerexin correlation with de-differentiation of cell?
Yes. So we have not seen, when we delete scleraxis, initially our thought was that in fact what is happening is de-differentiation. But now we find that it's not actually de-differentiation. Basically, these cells do have, they are specified as tendons, but they just do not elongate. But on the other hand, another projects that another postdoc in our lab works on is the role of TGF-beta signaling in tendons. And she has been able to show that when you delete TGF-beta signaling in tendons, these cells actually de-differentiate. So they become they take a somewhat progenitor form. Yeah, so we do have de-differentiation in tendons. We do not have any data so far to show that scleraxis is involved in it. We know that scleraxis is important for the cell fate specification, but we have not seen data that suggests that it may lead to de-differentiation if you delete it. Yeah.
Maybe upstream or downstream of scleraxis?
Yeah, so since we know that TGF-beta leads to de-differentiation, or deletion of TGF-beta leads to de-differentiation, and the way TGF-beta does its role is through the SMAD proteins. So one of some part of a project in our lab actually is working on SMAD proteins, and we have been able to show that it's possibly a likely link between scleraxis and SMAD that might lead to de-differentiation. Yeah. I think Utpal has a question.
Yes, ma'am.
Okay, so, okay, he has asked, which embryonic cell is typically responsible for tendon generation? Is it mesoderm or neuroectoderm, which also gives rise to adipose tissue? So what is the origin? That's a very good question, Utpal. So mesoderm gives rise to the muscle, and pretty much most of the mesoderm also gives rise to most of the skeletal tissue. Except there is this lateral plate on the mesoderm. So when you look at somatic stage, there is a lateral plate on the mesoderm that was thought to give rise to tendons, but we eventually found that there is a layer right next to it, which is called as the syndrome, and that is what gives rise. So if you look at it primarily, yes, it is coming from a part of the mesoderm, that gives rise to the tendon cells. Does that answer your question? Okay.
Anshul, you have something, right? If I'm not wrong.
No, I was at that time. You were off. You try to forget your question. No, I shouldn't say try. You forgot your question. What you had to ask.
Yeah, I'm growing old. You know.
Throw in some translation there. What happens to the translation state? So, well, since you brought it up, this is very, again, naive question. Is I understand mice, mouse model, they are nocturnal system, right? They work in the night, sleep in the day.
Yes.
Well, I shouldn't say we work 24/7, I'm sure. So let's go into, normal people work in the day and sleep in the night. So regarding this single cell RNA-seq in experiments that you have conducted, again, this is me just assuming that scleraxis, if I pronounce it right,
Yes.
the transcript or the protein levels are consistent throughout the day, or they have a dial pattern. Any idea about that?
So that's an interesting point. You're right. So mice indeed are nocturnal. So they're mostly active in the night. As far as tendons are concerned, a research lab in London has actually, they are big names in our world. They have shown that tendons indeed have circadian rhythms. So if you scan tendons using the electron microscope, say morning, and then you scan it in the evening,
yes.
You will see a change in the kind of collagen that you're seeing. At the genetic level, at the signature level, you do not see a change in the genes, perhaps, but you will see a change in the genes that are regulating this collagen structure. So for scleraxis, we have not seen, for our single cell RNA-seq, we have not looked at, or we have not identified any circadian rhythm patterns in there. But one of the reasons was also that we have tried to remove that variable. So all the experiments that we have done so far for single cell, I always do it at the same time every time. So we have removed that variable out of the equation. But you're right, tendons do have a circadian rhythm, and that mostly affects the collagen in the tendon. Yeah.
So the overall idea was a very, kind of a shotgun approach to see, hey, these are the cells which might be, sclerosis might be responsible in attracting these versus, the minus, the knockout mutants that you had. So, and then once you identify the next level approach, like you're saying that, you're going to do more functional studies and genetic studies to characterize that. Yeah, that's a pretty nice pipeline. Yeah, it was nice research. Very good. Yeah.
Thank you. Thank you. Same for you.
Yeah. Thank you.
Okay, ma'am. Once again, I thank you so much for such a wonderful session, ma'am. Now, I request Dr. Uppal Dongre sir to propose vote of thanks.
Am I audible?
Yes, sir.
Okay, thank you, Swati. I am Dr. Uppal Dongre. Welcome all for valedictory. By taking blessings from Tathagat Ambedkar, words of humanity, I would like to propose a formal vote of thanks on the occasion of this international webinar entitled Expert Directives and Experiences to Develop a Global Research Career in Biological Sciences. At first, I would like to thank management members of Param Pujya Dr. Babasaheb Ambedkar Smarak Samiti for their constant support, help, and motivation for organizing such informative webinar. A special mention to our respected principal, Dr. P.M. Siriya Madam, for being the catalyst that stimulated us to do our best and standing as pillars of strength. I would like to express my sincere thanks to Dr. Hema Menon Madam. But I am audible. Okay, sorry. IQSC coordinator of our college for her perfect logistic support and guidance. I would also thank our cheerful, supportive, and energetic HOD Madam, Dr. B.A. Mehere, for motivating us to organize such a wonderful webinar. Friends, how can I forget to appreciate the efforts of today's most distinguished, knowledgeable, enthusiastic, and renowned speakers, Ms. Snehal Raut, Mr. Sujeet Bhoite, Dr. Ansul Lokdarshi, Dr. Sunayana Mitra, and Dr. Depanwita Pal. I believe today we are blessed for having such expert faculties with us who shared their experiences and knowledge with the upcoming wealth of the nation, that is students. Friends, I think it is now 5 to 5:30 a.m. in Portland, and at this time, the lecture delivered by Dr. Depanwita Pal is really admirable and appreciable. Hence, thank you all speakers for sharing such wonderful knowledge with us. But this vote of thanks would be incomplete if I forget to mention the rigorous and continuous efforts taken by Dr. Devrat Begde sir and Mr. Rohan Thaure for organizing this webinar. Please give them a big round of applause. Their meticulously planned and perfectly organized events made this webinar a grand success. I owe a special word to express my deep sincere thanks and gratitude to all our teaching staff members for the smooth conduction of the webinar. I would also thankful to all dear student participants. Without their presence, this webinar would not have been successful. I would like to thank our non-teaching staff and Nishit Jain as well as Sachin Ramteke from TriStar Solution for providing the technical support. Please forgive me if I forgot to mention people who directly or indirectly helped us to conduct this webinar. Thank you.
Thank you, Utpal, for that.
Please continue.
Yeah, thank you, Utpal, for that comprehensive vote of thanks. I would, on my behalf, would like to thank everybody who have been associated with this venture. Thank you, Anshul. Thank you, Sunayana. Thank you, Sujeet, Snehal, and Depanwita. Hope to meet you again soon. Okay, if you can turn on your videos, then it would be nice to say bye.
Thank you. Thank you so much, everyone.
Sunayana already on the beach, right?
Thank you so much, sir, for giving us this opportunity.
Yeah, thank you.
I'm kind
Thank
of
you,
jealous.
Snehal.
You're right. She's already on the beach, you know.
Sunayana and you were on the same batch, right? Anshul?
Yes. Yeah. Very close colleagues.
So I forgot to. I actually forgot to introduce to you through Sunayana, right?
No, I saw her. I know we were kind of chatting separately. We're just trying to greet each other. Yeah, that was on.
Yeah. Right, and for those who are watching online on the YouTube channel, the feedback link has been posted in the description, so they can go and click the feedback and fill it to get the e-certificate of participation. So thank you all. Hope to see you soon again.
Yes.
Yeah.
Thank you so much.
Thanks everyone.
Bye.
Yeah. Bye-bye.